Welcome back from lunch. Hope you had a good one. Hopefully more people will trickle in. It is truly an honor for me to introduce Dr. Patricia Darrah. She received her PhD in micro and immuno from Temple University in 2001, then joined the Robert Center Cellular Immunology section at NIH for the Vaccine Research Center, where she began using flow to understand vaccine-mediated protection. She became a staff scientist in 2009 and transitioned to TB research in non-human primates, becoming head of the VRC tuberculosis unit under Bob Setter and Mara Roeder in 2016. You were in for a treat for an exceptional talk. From a personal point of view, Darrah probably didn't know me from Adam 4 months ago until I emailed her and said, we want you to come to FlowTex, because myself and a few other people heard her talk at Cyto. And depending on how you view sessions at other meetings, sometimes they're really good and exciting and you just, this is a great talk. Other times you grab your cell phone and check your email, to be honest. Well, I was on the edge of my seat. And for me, not saying anything with all due respect to the program committee of Cyto, Patricia's talk at Cyto was one of the showstoppers in my mind. I walked out of Cyto thinking, I've just heard an incredible seminar. So with that, it is an honor to introduce Patricia Darrah, talking about correlates of protection against TB after immunization in macaques.
Wow, thank you for that great introduction. I hope I can live up to that. Thanks for the opportunity to speak to you today. It's a pleasure to be here. So as Dave said, my name is Patricia Darrah. I head the TB unit at the NIH Vaccine Research Center under Bob Setter and Mario Roeder. And the work I'll show you today has been a collaboration with Joanne Flynn at the University of Pittsburgh. So this is going to be a pretty data-driven talk, but the main technology that I've used to characterize the immune responses in this preclinical model has been flow cytometry. Okay, so tuberculosis is the deadliest pathogen in history. It's estimated that 1/4 of the world's population is infected with TB. Each year, there's 10 million cases of active TB and 1.5 million deaths. Sadly, in 2020, we saw our first uptick in more than 20 years in TB-related deaths because of the COVID pandemic and because of limited access to healthcare and diagnosis. Drug treatment against TB is extensive. It's a 4-drug regimen that needs to be taken for 6 months, and it's not well tolerated. Therefore, there's low compliance and low completion rates, and that directly results in a rise of multi-drug resistant strains of TB that are even harder to treat. So drug treatment is not really a cost-effective option or an option that's available to everyone, especially in low-resource countries. What is cost-effective is a vaccine. And in fact, there is a vaccine against TB. It's called BCG, and it's over 100 years old now. BCG is a live attenuated strain of Mycobacterium bovis that's given intradermally in TB endemic countries at birth. BCG protects infants against severe and disseminated TB, but as its immunity wanes, it fails to protect adolescents and adults against pulmonary TB, which is the most transmissible form. So just a few words about the bug and the infection. Infection with the bacteria, Mycobacterium tuberculosis, occurs person to person through the air. The bacteria infects alveolar macrophages in the airways and then goes on to cause disease in the lung. And that disease is characterized by these granuloma structures, these immune structures that act to try to contain the bacterial infection. Vaccines against TB can take several approaches. They can try to just reduce the disease severity or to try to prevent person-to-person transmission, or you can try to have maybe a loftier goal, which is to prevent infection at the start and prevent this early infection of macrophages and thereby prevent all the downstream events of active TB, latent TB, or reactivation. Either way, most vaccines against TB focus on cellular immunity. So the goal is usually to elicit antigen-specific T cells that are going to recognize these activated infected macrophages in the lung and to kill the bacteria inside of them. So this slide summarizes what we think of as the key requirements for a T cell vaccine against TB to be successful. And those include the magnitude or the frequency of that T cell response. And those could either be conventional T cells, CD4s and CD8s, or these unconventional or donor-restricted T cells, such as gamma delta cells, mucosal-associated innate T cells, or NK cells. We also think about the quality, meaning the phenotype or the function of those cells, including cytokine production or maybe memory markers or chemokine receptors. Then the breadth of the response, the diversity of different types of antigens recognized. And finally, the location of that response. In other words, what is the anatomical location where those T cells need to be in order to exert an effect upon, on the infection at the time of infection? And in the case of TB, that area is the lung. So from a vaccine delivery perspective, we wanted to ask, how can we generate a high frequency of durable antigen-specific T cells in the lung? So the approach we decided to take was to look at different routes of administration of BCG, and those included either direct aerosol delivery of the BCG vaccine into the lungs. We looked at the standard intradermal BCG vaccination, which we expected to target just peripheral lymph nodes and possibly spleen. And then we wanted to investigate intravenous delivery or systemic delivery, which we thought would target several organs such as the lungs, spleen, as well as generate a circulating reservoir of memory T cells. So this is the experimental design that we set up in rhesus macaques several years ago now. So animals were vaccinated with BCG intradermally at the standard human dose of 5 × 10⁵, or they received a 100-fold higher dose of 5 × 10⁷, also intradermally. So we had ID high and ID low. At the higher dose of the vaccine, we compared 3 different routes. So ID, aerosol, or intravenous. And then finally, we had a group that combined a peripheral and aerosol route. 6 months after vaccination, we challenged these animals with virulent Mycobacterium tuberculosis, and we monitored their disease both by longitudinal PET CT scanning and also by quantifying the mycobacterial burdens at the time of necropsy or at the end point of the experiment. So I'm going to jump right to the protection here and show you on the slide. On the left, there's a graph showing the total CFU in the lung. And on the right, you'll see these PET CT scans. These are individual macaque lungs, where the orange is areas of radioactive FDG or inflammation and indicates TB disease. So in our unvaccinated animals, we typically see 3 months post-challenge about 10 to the 6 CFUs in the lungs and pretty severe disease. These rhesus macaques are really, really susceptible to TB. Compared to those unvaccinated controls, the standard human dose of ID, or this ID low group, confers about a 1 log reduction in bacterial CFUs compared to the controls. If we increase the intradermal dose 100-fold, we can further confer a 1 log reduction in CFU compared to the low dose ID. You can also see a reduced amount of FDG activity in these scans. Kind of to our surprise, the aerosol group didn't seem to do much beyond what the low dose ID did, and that's what we powered the study against. So these animals didn't really do too well after mycobacterial challenge. However, we had this striking result when we looked at the intravenous BCG group. So let me just focus on those a little more. So these are the 10 animals that were vaccinated with IV BCG and then challenged. One of the animals in this group was completely not protected, and here's its level of disease. 3 of the animals had fewer than 50 CFUs, and we considered them protected based on our criteria. And then 6 of the animals had no evidence of disease whatsoever, so no mycobacterial CFUs and no inflammation on their PET scans. And so we classify these animals as sterilely protected, or in other words, we had prevented TB infection in these animals. We used 2 large flow cytometry panels to assess the immune responses in these animals after vaccination, and we looked in the bronchoalveolar lavage, or the bowel compartment, as a surrogate of the underlying lung tissue. And what we wanted to ask in this first panel was just simply, does the cellular composition of bowel change following immunization? So this panel has basic lymphoid and myeloid subsets, always heavy on the T cell subsets, and then some APCs, macrophages, dendritic cells, B cells, and K cells. And we just asked, does the composition of the bowel change? So the first thing we noticed when we started doing these experiments was that compared to the other vaccine groups, the number of cells that we would harvest in these animal lavages in the bowel was dramatically increased only in the animals that had received IV BCG. So this is pre-vaccination, 2 weeks, 4 weeks, and up to 16 weeks later. So these cells went up 10 to 50-fold. By flow cytometry, we could easily determine that this increase in total cell number, shown here in orange, was really driven by this dramatic increase in T cells, shown here in blue, which was either not visible or less visible in some of our other vaccine groups. Interestingly, the number of macrophages in green remained generally stable over time throughout the vaccine monitoring phase. We can look at this data another way. Here I'm showing you pie charts and how they're composed of the different leukocyte subsets in the bowel. And generally in these animals, the bowel is composed of about 75% alveolar macrophages. So those are shown here in yellow. So before vaccination, about 75% macrophages, less than 25% T cells. If the animals receive either dose of intradermal BCG, those proportions don't change appreciably, a little bit of an increase here in the high dose ID. In our 2 aerosol groups, we saw an increase in T cells at 8 weeks is what I'm showing you here, but 4 to 8 weeks after vaccination, where about 50% of the cells were T cells in these animals. But by week 16, those proportions had returned to what those animals looked like prior to vaccination. In our IV animals, though, we saw this really dramatic difference. So here by 8 weeks, more than 80% of the cells here are really made up of T cells and many fewer bowels. And again, that's because of the increase in T cell number. And that inversion was really maintained for at least 4 months, which is the last time point that we looked at these animals before their 6-month challenge. So the way I like to think about these data is as an effector target ratio. So if you're a person who believes that effector T cells are going to be responsible for activating infected macrophages to kill the bacteria inside of them, then this effector target ratio might be conducive to that or facilitate that. For the T cell aficionados in the crowd, we also looked at what kind of T cells were increasing in these animals. So now these pie charts are only the CD3 positive cells. So CD4s in dark blue, CD8s in light blue, and then in yellow and red, these 2 subsets of innate-like T cells, gamma deltas and nates, which have been shown to have some contribution to protection against TB. So we saw this interesting finding where gamma delta cells and nates were really increased in our IV animals, also here in the ID, high-dose ID animals. At week 2, that's fairly transient, coming down by week 4. And by the time of challenge, week 24, what we're left with is really a larger proportion of CD4 T cells compared to CD8s relative to what we saw pre-vaccination. The second flow cytometry panel that we used was to look at antigen-specific T cells. So here we really wanted to ask whether the route of administration of the BCG affected the antigen-specific T cell responses, again, in the bowel compartment. And this panel was made up mostly of T cell markers, some chemokine receptors that help define different T helper subsets, and then markers of antigen specificity and activation. So upregulation of CD69, CD40 ligand, and then also Th1 and Th cytokines, including interferon gamma, IL-2, TNF, and IL-17. So we chose these cytokines because of some literature that is and what's known about the immune response against TB. And I won't go through this in great detail, only to say that it's known that CD4 Th1 cells and the production of interferon gamma and TNF are really critical to protection. We know that because in humans, if you have an interferon gamma or an IL-12 defect, you're being treated with anti-TNF, or if you're depleted of T cells, like people living with HIV, these people are exquisitely susceptible to tuberculosis. We also know that IL-17 from CD4 cells helps drive T cell and neutrophil recruitment into the lungs, and CD8 T cells have been shown to contribute to protection both by maintaining latent TB, so maintaining TB within those granulomas, or also by direct killing of infected macrophages. So that's why we're focusing on these cytokines in our antigen specificity assays. So this graph now shows the number, so the absolute number of antigen-specific CD4 T cells reacting to MTB antigen and producing any combination of interferon gamma, IL-2, TNF, or IL-17 before and in the 4 months after vaccination. So if the animals had received low-dose ID, this is the immune response that they had at the site of infection. So there's an increase, less, maybe a half log or log increase compared to pre-vaccination. So compared to this standard regimen of BCG, the increased dose of ID BCG could increase the CD4 response almost a log. The aerosol groups had a similar looking kinetic of CD4 T cell responses compared to the high dose ID, but the IV animals really had this dramatic and robust CD4 T cell response that was, at least 2 logs higher of antigen-specific CD4 T cells right at the site of infection. Additionally, moving to the CD8 T cell responses, the intravenous BCG really seemed to be uniquely capable of driving a CD8 T cell response compared to our other vaccine groups. If you know me, you know that I've spent a long time studying multifunctional or polyfunctional T cells. And so when we looked at the CD4 T cells and the quality or the combination of different cytokine combinations that those cells were making at the peak of the response in the bowel, we really couldn't appreciate differences between these groups, despite that really large difference in magnitude. So clearly, these don't look qualitatively different, at least by the measures that we've picked out and curated to put in our flow cytometry panel. So I told you before that MTB infects alveolar macrophages and then makes its way into lung maybe 7 to 10 days later. And I've shown you that the T cells in the bowel increase, the antigen-specific T cells in the bowel increase, but we also really wanted to know whether the lung tissue was similar to what we were seeing in the bowel, and if we saw an increase of T cells in the bowel. So we applied this intravascular CD45 staining technique, where you inject a fluorochrome-labeled anti-CD45 antibody into the animals intravenously just minutes before euthanasia. That antibody then quickly circulates and labels all the leukocytes in CD45 positive cells in the blood, but it fails to stain those cells that are kind of protected in the tissue. So we have tissue versus blood based on CD45, and we also stain with CD69, which is a marker of tissue residency for the lung. On the right, I'm showing you different lung lobes from these individual representative animals that have received the same dose of either ID, IV, or aerosol vaccination. So this is 1 month later, and I think it's pretty obvious that the IV drives this really huge number of cells that are CD45 negative and CD69 positive, so tissue-resident lung cells compared to the ID animals and the aerosol animals, which show a bit of an increase, but nothing this dramatic. So in summary of this first part, I've shown you that the IV BCG is more protective than ID and aerosol, even at the same dose, that it substantially alters the proportion of T cells to macrophages in the bowel, and that it elicits the highest number of TB-specific CD4 and CD8 responses in the bowel and also in the lung tissue. So if you're not a TB person, I'll just stop for a minute to say that the study that I just showed you was really exciting because it really showed an unprecedented level of protection in a very susceptible rhesus macaque model. And because of that, it really provided us a benchmark to try to study and learn about immune correlates and mechanisms in this preclinical model. And those are the two types of studies that I'm going to show you next. So to look for correlates, we weren't able to do that in the first study, and that's because out of the 10 animals that got IV BCG, only one of them was really not protected. So we didn't have the power to ask what's different in the immune responses between the protected and unprotected animals. And in fact, you have the most power to discover immune correlates when you have a 50% protection rate, and you can look at what's different about those that are yes or no protected. So to try to force this in a study, we set up the following rhesus macaque study where we vaccinated 34 non-human primates with half-log increases of IV BCG between 5 times 10 to the 4th to 5 times 10 to the 7th. And this highest dose was just a smidge lower than what I just shown you in the previous published study. 6 months later, we challenged these animals with Mycobacterium tuberculosis and measured their bacterial burdens 12 weeks later. So again, the rationale of this experiment was to use the varied IV BCG dose to generate a range of immune responses to which we hoped to correlate different protective outcomes. And in fact, we were able to generate a range of immune responses. These graphs show the antigen-specific CD4 and CD8 frequencies of cells producing these cytokines, and they're grouped by vaccine dose. So as the BCG dose increased, you can see that the median levels of these cytokine production increase, although there's variability amongst the individual animals, which are shown in the light green lines. We saw something similar in CD8s, although the CD8 responses top out at about 10%, where in the bowel of these animals, we routinely see greater than 60%, 50 to 60% of the memory T cells in the bowel responding to our BCG or TB antigens. We saw similar dose responsiveness in the PBMCs, although again, these frequencies are 20-fold lower than what we see in the bowel. But again, the highest doses of BCG drove the highest responses in the blood. So moving to the protection, again, we saw the expected level of bacterial CFUs at necropsy, so 10 to the 6th in our unvaccinated animals. So compared to these unvaccinated animals, we saw a wide range of protection across our whole range of IV BCG doses. So at the highest dose, which was similar to our historical controls, the study I just showed you, we saw a similar level of sterile protection, about 75%. However, across all of our doses, even this lowest dose, we saw one animal that was sterilely protected. So in general, there was association between dose and higher levels of protection. Still, across all the animals, we had 18 out of 34 animals protected, and 17 out of 34 of these were sterilely protective. So every dose, we had protected and unprotected animals, which we thought set us up nicely for looking at correlates. So because we had really collected a huge amount of longitudinal immune parameters in the blood and bowel before and after vaccination, we wanted to take a more integrated and perhaps less biased approach of looking for immune correlates. And so we decided to take a systems analysis approach. So again, most of the data that we had generated stemmed from these two even larger flow cytometry panels that have been developing as we were able to use more and more colors. But again, one of the panels was really based on cellular phenotypes, trying to understand the difference, how the cellular composition might change in the blood or the bowel after vaccination. And the other was an antigen-specific panel looking at CD4 and CD8 T cells, innate T cells, their phenotype, their cytokine production, or their cytolytic function. We also had measured plasma cytokines using a Luminex assay after vaccination, and we took a large number of measurements of antibody titers to a variety of different MTB antigens. So of all that data, we had about 900 immune features. And from that, we tried to transform this data to simplify it a little bit. So we calculated areas under the curve from these longitudinal parameters to distill this data down to what became 68 bowel parameters and 83 blood immune features that we wanted to try to correlate to protection. So we fed this all into a multivariate systems analysis. And when I say we, I mean we did this in collaboration with Chauncey Wang, who was in Doug Laufenberger's lab at MIT at the time. So to start, we defined protection here categorically as either yes or no. The first analysis that Chauncey did was a lasso PLSDA analysis, which is similar to a principal component analysis, where he asked that if he just used the parameters from the bowel, whether he could discriminate protection. And in fact, he could. So you can see protected animals in orange, unprotected animals in maroon. Furthermore, he was able to simplify this and predict protection using a minimal signature of just 4 lasso-selected features, and those features are shown here. So these 4 features are accounting for most of the variability across this axis, and those 4 alone can predict protection. And those included 2 of the CD4 cytokine-producing populations, those making interferon gamma and TNF, those making IL-17 and TNF, and then the number of NK cells in the bowel, and one of the antibody measurements, IgA, to the PPD antigen. This model was fairly robust at predicting protection. It correctly categorized 16 out of the 18 protected animals and 13 out of the 16 unprotected animals. When we moved to do the same analysis in the blood, we found that the blood immune features were less robust at discriminating protection, as you can see by these larger ovals and greater overlap. Furthermore, 6 features here were required to minimally predict protection, and those happen to be two of the parameters that we measured from the plasma assays, and then 4 of the T cell features, which seem to be associated with effector memory or cytolytic activity. This model, although it predicted 17 out of the 18 protected animals, was less good at predicting non-protection, leading to a significantly lower correlation coefficient compared to the bowel analysis. From there, Chauncey did this immune network analysis. So super busy, but let me try to just point out a few important take-homes from this. So in this analysis, those 4 lasso-selected features are represented here by the 4 central orange nodes. And then we asked what other immune features in the bowel that we measured were co-correlated with those 4 central nodes. And there were a lot of them. Okay, everything here, the correlation coefficients are shown in green. So the darker the green line, the greater the correlation. So if we just kind of look at this first node, the number of NK cells, so what's in here? We see some CD4 populations. We see a lot of things that also express CD8 alpha, CD8 cells, MATE cells, V gamma 9s. But if you see a really dark green line like this, and you see a bunch of them here, what it means is that the number of CD8 cells would have been almost as predictive as the number of NK cells in predicting protection. In other words, a lot of what we measured in the lab would have predicted protection. So that was pretty interesting. The other nodes here I won't belabor, other than to say that the antibodies kind of co-correlated with other antibody measurements, and the CD4 nodes correlated with a lot of the other cytokine combinations production by other CD4 T cells. In contrast to the bowel, the PBMCs gave us this sadly puny immune network. So only 2 of the 6 lasso-selected features really were co-correlated with other immune features. One was one of the gamma delta cells, a cluster of gamma delta cells, and the other were CD4 cells that were effector memory-like. But I think you'd agree that compared to the blood, the bowel really gave us this, revealed this highly coordinated immune compartment following vaccination. Okay, so in that analysis I just showed you, we hadn't considered the effect of BCG dose in those analysis, but I did already give you examples of how the dose was driving our data. So the higher the dose, the higher the immune response, the higher the dose, the higher the protection. Basically, we want to know, we needed to know what wasn't a factor of dose and what really was a correlate of protection. So were we able to identify dose-independent correlates? To do that, Chauncey created this nested mixed linear model, and the data from the blood and the bowel are shown in these two volcano plots. And the way to interpret these plots are to focus on the top right-hand quadrant, which show you the immune parameters that are most strongly and most significantly associated with protection. So first off, the blood showed us a few parameters here, and based on the scale, they were less strongly predictive of protection. Whereas in the bowel, a lot of the features that we measured withstood this dose correction. And in fact, 3 of the 4 lasso-selected features that we already talked about are still here. Interestingly, all of the antibody measurements dropped out of this analysis, suggesting that those might be more associated with dose rather than the actual protection. And if we just look at the top of this curve, what we see here are that the strongest predictors of protection ended up to be plain old CD4 cells, either making interferon gamma, or the number of CD4 cells producing any combination of gamma to TNF or IL-17. So at least for IV BCG, I think these data strongly suggest that CD4s are a correlate of protection. And what we wanted to know then was, are these CD4s a mechanism of protection? And spoiler alert, they are, and I'll show you that next. But let me show you one more thing that we're trying to do with this data we're just starting, but that's kind of cool, I think. So up till now, the multivariate analysis, I told you we started by creating these normalized areas under the curve to simplify the longitudinal data. But by doing that, we basically ignore all the longitudinal data and we lose that temporal information. So we're trying to do this analysis called data stream clustering. So if you think of your longitudinal data over time as a stream of data, and this is your cartoon of that data, you can appreciate that if you mixed all these clusters together, you could find 3 clusters: gray triangle, red hexagon, blue circle. Right? But what you wouldn't be able to do is know that the triangles only come in the beginning of the data, and then there's this alternating pattern, red, blue, red, blue. And that's what data stream clustering does. It tells you not only at what point the clusters are prominent, but also the probability of one cluster transferring to another. So if you're a gray triangle, 2 out of 3 times you're staying a gray triangle, but 1 out of 3 times you're becoming a blue circle. So how does this apply to our data? So now in the UMAP space, we clustered all our bowel data and we looked at not only the individual animals, but the time points. So you can appreciate 6 clusters here, but now each dot on this plot is a single animal from a single time point that we measured. If we go to all of our immune features and we try to order this heat map by clusters, here are the clusters in this column. So red is number 1, orange is number 2, light green is number 3. Within each one of those clusters, there's some animals that are protected in orange, some animals are not protected in maroon. And for each clusters, the proportion of protection is different. So how do we make sense of this? We now add in the temporal data, we can make a plot that looks like this, a wave plot or a ribbon plot. And so on this plot, I'm showing you only the protected animals. These are the animal IDs of those protected animals, and these are the time points after vaccination, so 0 to 3 months. The colors represent the clusters, and the clusters are connected by these ribbons or waves that tell us how the clusters transition one to another. So in this experiment, or in these animals, all the animals started off in this red cluster 1, transition at 2 weeks, aqua and light green, but then they really end up strongly in these green and then this purple cluster. So by 8 weeks and out to 12 weeks, these animals are staying in this immune cluster. And so we were really curious to know whether unprotected animals looked exactly the same or might look different. So we were excited to see that these animals did look quite different, except at time point 0. So that's good because it tells us that there's nothing inherently different about our animals at the start of this experiment, but they really follow a different path. They hang out in this orange and light green, and they never really make it to these purple clusters like the protected animals do. And I don't have all the data about the signatures that drive these clusters, but that's what we're working on right now. So I'm going to leave it at that, but I wanted to show this slide because they're pretty. So in summary of this analysis, the immune measurements from the bowel predicted protection better than those from the blood. And if you're not a person who thinks about clinical trials, I point this out because in clinical trials, usually people are collecting blood. It's the easiest and most accessible and cheapest compartment to sample from which to sample. So we would really advocate for trying to sample a subset of patients to look in their bowel for immune correlates. So protection after TB was influenced by dose, but despite correcting for dose, we were still able to identify multiple immune features that were correlated with protection. And those were some of the frequency and numbers of CD4 T cells expressing cytokines, and then some of the CD8 alpha expressing cells, NK cells, and possibly some of these conventional or unconventional T cells. But again, the correlates may not be mechanistic. So we really wanted to know, is this immune correlate, like what are the immune responses that are mediating protection after IV BCG? So to do this, we undertook a large T cell depletion study in rhesus macaques using available reagents from the NIH non-human primate reagent resource. We used anti-CD4 or anti-CD8 alpha. In non-human primates, the anti-CD4 is pretty straightforward. It's going to target these single positive CD4 T cells, as well as this 10% or so of double positive CD4, CD8 T cells that are seen in monkeys and to a lesser extent in humans. The CD8 alpha gets a little bit more complicated. So anti-CD8 alpha is going to deplete any cell that's expressing CD8 alpha. So that turns out to be not only your classical or conventional CD8 T cells that express CD8 alpha beta, but also any cells that express the CD8 alpha alpha homodimer. And that includes NK cells and some of these innate T cell subsets like gamma deltas, mates, and iNKTs. So this anti-CD8 alpha depleting antibody is a really broad depleter as opposed to the CD8 beta, which would more specifically target your conventional CD8s. So in this experimental design, we vaccinated most of our monkeys except for a couple unvaccinated controls. Here, animals received a high protective dose of IV BCG. 5 months later and 1 month before challenge, we infused them with either control antibodies or saline or antibodies to deplete either CD4 or CD8s. We then challenged them with MTB and asked whether the protection that we expected to see from IV BCG was abrogated when we got rid of either of these cell types. So in the next couple slides, I'll just show you quickly that these animals had good immune responses. I'll show you that the depletion worked, and then I'll show you the protection from this experiment. So as I showed you in earlier slides, but now I can show you with a bunch more animals, after vaccination, we see this huge increase in T cells in the bowel after IV BCG vaccination. And that causes, again, this inversion of macrophages to T cells in these guys. So, and not only that, we see again in this experiment, this burst of innate T cells, the V gamma 9s and mate cells, that's transient. And we end up with more CD4s and CD8s. So similar to what I've shown you before, these animals also had the expected antigen-specific immune responses. These are the numbers of antigen-specific CD4s. We're about log higher than the CD8s. And now I'm adding in the V gamma 9 cells, which we now measure the cytokine production from. So from historical perspective, these animals were behaving just as we thought. So a month before challenge, we started infusing the depletion antibodies, and we diffused those antibodies throughout the challenge phase until the end of the experiment. And this slide shows you the extent of depletion after we infuse the antibodies. So on the top, you can see in red the frequency of CD4 T cells before or after depletion in animals that receive the anti-CD4 antibody, right? So compared to the controls. And on the bottom, you can see the percent of CD8 T cells in blue after the infusion of the CD8 alpha antibody. So this antibody works great in the blood. You had this extensive and sustained depletion throughout the challenge phase. We were also curious in looking at tissues because if you know anything about these antibodies, people often say, well, it's easy to deplete in the circulation, but it's hard to deplete within the tissues. So we looked in the bowel just before and after the first infusion of the antibody. And here we saw about 76% and 91% of CD4 and CD8 T cells depleted after a single injection of depleting antibody. And also at the bottom, I'm showing you the double positive T cells expressing CD4 and CD8 were depleted to about the same extent as the single positive cell in the same treatment group. We also had an opportunity to look at the lung tissue. So now this is before infection. This is at the end of infection. So these are the animals that are actually challenged. So at the end of this experiment, we measured CD4s and CD8s in the lung tissue and saw about a 90% depletion. And this was really important because I keep showing you that the T cells really are increased in the bowel and the lung tissue after IV BCG. So we wanted to make sure that these antibodies were working. So what did the protection outcome look like? Can these animals still protect without CD4 or CD8 T cells? So these are the outcome data. Our unvaccinated controls up here, really high disease. Compared to those, the animals that received IV BCG in green, but were not depleted or were control depleted, had about a 3-log reduction in bacterial CFUs. 3 of those animals were sterile. So compared to these, animals that received anti-CD4 depletion, we saw completely abrogated protection. These animals were as bad, if not worse, as our unvaccinated controls. And these data really support CD4 T cells as not only being a correlate, but a mechanistic correlate of protection. Our CD8 alpha group looked a little bit different. Some animals here did really bad, but we sort of had the split phenotype with 4 of the animals doing perfectly fine and being sterilely protected despite the CD8 alpha depletion. So either it's different in different animals or the level of depletion wasn't as complete in these animals as in these. So there's definitely an effect here, but maybe not as clear cut as the anti-CD4. We also looked at CFUs in the lymph nodes, and we do this because it's a measure of dissemination of the TB disease not being controlled in the lung and spilling out into the lymph nodes. And here you can see that both the CD4 and CD8 have high lymph node CFUs. The CD4 depleted animals have higher CFUs than the CD8, but both of them are significantly higher than the IV BCG animals, which really do a great job at containing this infection in the lung and not letting it spill into the lymph nodes. So to summarize these studies, I've shown you several studies, the first or the second really being the correlate study, the dose ranging study, where I hope I convinced you that we were able to find cellular immune responses in the bowel that correlated with protection, and that the correlates in the bowel were better than blood at predicting protection. In our T cell depletion study, we've confirmed a primary role for CD4 T cells in IV BCG mediated protection. And we also showed that the CD8 alpha impacted protection in a number of the animals. And again, those cells could include these CD4 T cells, the NK cells, MATES, and gamma delta cells. And so we're repeating this experiment with a CD8 beta depleting antibody to try to deconvolute and understand these results a little bit better. So in terms of broader implications for vaccine design, we really believe that the IV delivery here is the key to generating a high frequency of lung resident T cells. And people don't like IV delivery, although it's actually being done for some malaria vaccines and also monoclonal antibody infusions in a number of trials for malaria and for HIV right now. So if it turns out that CD4 and CD8 T cells are required, then that really makes you think about what type of vaccine modalities would be appropriate for TB. So we're using a whole live attenuated vaccine. It's generating both CD4s and CD8s, but some vaccine types, like a subunit vaccine, for example, are not very good at making CD8s. So if you need those CD8s, you're going to need a viral vector, maybe a good mRNA vaccine, or a whole organism vaccine. And another aspect of the whole organism vaccine is really we're using a whole bacteria. BCG has got about 4,000 proteins. So, you know, it's not a virus, it's a bacteria. There's a lot of antigens, and we're probably getting a broad breadth to a lot of different antigens. So we're in the process now of trying to map some of these antigens and understanding if there's any immunodominant antigens that might be able to be fed into a subunit approach that would be safer than vaccinating with a live bacteria intravenously. Still, it worked very well. Okay, so with that, I'm going to conclude and just acknowledge the people who did the work. This is my small group at the VRC. Again, all the challenge data was done by at the University of Pittsburgh with Joanne Flynn's group. And then Chauncey did all the systems analysis here. And we've had funding from the NIH and also some funding from the Gates Foundation. With that, I'll stop and take questions if you have any. Thank you.
Any questions? Oh, there's some online ones too. Okay. Might be off turn. Make sure you can turn that one on. Tap it. Or go Leroy real quick.
Do you hear me? Is it working?
No.
Yeah. It is. Well, thank you very much. It was really interesting to see these vaccination strategies and how you evaluated the immune response. I was wondering, through all your data, you see that even at the low dose vaccinations with BCG, most of your animals show pathology in the lung, but then you have one, like you said, one rhesus macaque that is sterile.
Mm-hmm.
Can you say something about maybe the underlying mechanisms that even that you see such a difference in outcome based on?
Yeah, in some of the experiments, it's been interesting that we see this really binary outcome. They're either really bad or really protected. And we've seen that in a few experiments. More recently, we've seen a more range of middle lying animals, but that's what we're trying to get at with the correlates, right? So what we've done now based on that experiment is to really understand a 50% dose where we can give all the animals the same dose. Then we don't have that confounding variable and try to look at correlates from there. But yeah, there's nothing. It's been hard to dissect a signature that isn't just driven by the high dose of IV BCG and to understand what's different about that one guy that did really poorly. So yeah, I don't know.
Is it maybe known that it has something to do with the genetics of that particular monkey?
Yeah, I think there's a lot of that. If this is a non-human primate model, it's outbred. Animals come from different places and have different backgrounds. They're born, bred in different areas, so they have different exposure to NTMs, non-tuberculoid mycobacterial species. So they have some pre-existing responses. They're not mice, and so we started to track things like just MHC typing. And, it's kind of hard to know what their mycobacterial exposure was, but we do pre-screen them. So yeah, I think microbiome analysis is another thing we've thought about a lot. So yeah, how they respond to the vaccine is maybe something that's genetic that we talk about it a lot, but we don't know.
Thank you.
Hi, Tricia. Such a beautiful study.
Thanks.
And really, really nice presentation. I'm just curious to know if you have any plans or you thought about how long this response lasts. If you have like another time points to challenge.
Yeah. So all of our most of our challenges have been at 6 months post-vaccination, and that's really a cost factor of keeping these animals on study. And we have had not on purpose. We have vaccinated animals a couple months later, and it looked like, maybe we had a slightly lower efficacy, but we don't know. We haven't done those memory time points. So we haven't done something out to 2 years. Right now, we've been focusing on like the lowest, like the minimal dose that we might need, trying to dose back, because I don't think we expected to have a protection at such lower levels. So we were trying to think about how we could really develop this clinically. And of course, like bringing down the dose was one of those, the first thing that we wanted to try. So we don't know. But let me just add that we did measure responses out, pretty far. So we can measure responses. I think I've measured out probably to like 2 years, where they're certainly not as high, but they're still there. But, you could think about boosting too, which is doable. We've boosted monkeys with multiple shots of IV BCG and seen that we can keep those responses level for longer.
Okay, thank you for the interesting results. So I have 3 questions. So since BCG is already, so in our country, in the Philippines, it's part of the neonatal vaccine program. So how do you address, so if you will be pushing through this, the ethical issues, especially these are children, so the potential to, activate the infection instead of protection. And then next is, in your study, did you ever have any severe adverse events on these animals? And your plan on doing this as a booster, as you mentioned?
Yes. So where you would want to give this vaccine is definitely places that are giving ID BCG at birth. This is not something that would be replacing neonatal vaccination. It's something that we would think about doing as an adolescent boost. So we have done experiments where we've boosted ID vaccinated animals, and it seems like we're getting, we're not limiting the response, we're getting the booster that we had wanted. So I think it would work even in the context of neonatal vaccination. I think in terms of adverse effects, we monitor that carefully. We do see transient splenomegaly. There's an influx of cells in the lungs and the spleen, and that's really, people do worry about that. But it seems to be, in the first couple months, we see it goes away. We haven't had any, nobody's died. We haven't had any major effects. We see transient liver enzymes increase, transient CRP, no temperatures. If we give multiple doses, we start to see temperatures, but we haven't seen any really serious side effects. So Was that it? What was the third thing, real
Yes.
quick?
Thank you.
Plans for development? I'll have
Sorry.
to see.
Yeah, thank you.
We could
I-
do online,
Yeah.
online people, one little shot in here, because we got 3 on the online as well. What about follicular T cells serving as reservoirs for this disease? Would it work the same as it does for viral reservoirs?
Follicular T cells being a reservoir for MTB. I'm not, I guess I'm not aware of MTB being infecting those cells. Mostly macrophages, neutrophils, DCs. So I have a lot to comment on TFH.
Yeah, good talk. I have a couple of questions. Sorry for.
No, I'm
Right.
not.
So I have first question is that how you decide like low dose or high dose? It's like dose is like depend on the immune response, because in the you have high dose and still you have some some infections. But if you increase the dose further, can you do this or that's how you decide like doses?
I think the question is more if you have a little bit of infection, what happens down the line? So we're euthanizing these animals at 3 months, right? We're stopping the experiment, but we don't really know whether those 50 bugs that are there would go on to be awful disease or whether they would be killed off over time. So even though I say, oh, we had protection, there was less than 100 bugs, I'm not sure. You might need sterile protection, or it might be, it might be good enough to reduce severity of disease, and that would help prevent transmission. So, sterile infection is a really high bar that might not be required in the field. I think, reducing infection by 50% could have, a monumental global effect on reducing disease transmission. So this rhesus model is a little bit different than humans. They're more susceptible. So they seem to do really poorly if you don't protect them completely. But I think in humans, the bar might not be that strict.
Yeah, and second question is like you depleted CD4 T cell and CD8 T cells, right? In monkey, right?
Mm-hmm.
So I see like monkey is very big animal. So how much antibody you need for that depletion, and how
Yeah.
many doses you have injected?
Yeah, it was a lot. It was an expensive experiment. I think we did 50 mg per kg, and it was every 2 weeks. So starting at a month before challenge, and then for 2 months after. So 3 months altogether. So 6 injections, I guess, per animal. So yeah, it was.
It's like mg per kg or some how you like?
How did I deliver it?
Yeah, no, the quantity like mg per kg of weight or like how you decide?
50 mg per kg. Is that what you're?
Okay, 50 mg per kg.
Yeah.
Oh yeah.
Yeah, and that reagent is from the NIH
Yeah.
resource, so it's expensive, but it's not doable. I think it's a great
I have
resource.
one more question. This is personal. So I recently got tested. So I found like TB positive. So I never have in my life any like symptom. So how you see this one? So
Skin
later
test? Where's
I got,
Marsha? We need Marsha. A PPD test?
that interferon gamma test. And interferon gamma release.
I think you saw some TB somewhere. Did they give you a chest X-ray?
So and then I got like treatment for maybe it was 4 months.
Yeah. How was it?
So I don't know right now what is the situation, but I never have any issue. I never had any like.
Were you in
any
prison?
symptoms. I'm from India. That may be the reason
Yeah.
because of that.
Yeah, I mean, it's not completely absent in this country. I mean, there's been outbreaks here, which is what I was talking to Marsha about last night. That's why I brought her up.
Diana.
Patricia, thank you so much. That was a very interesting talk. Just curious, in your depletion system or study, you were seeing that the increase in CFUs. Just curious about the granuloma structure, if it was also changing and how that was impacting as well.
Yeah, that's a big question. I'd like to know. So we're trying to look histologically at some of those animals in the lungs. If there's no infection, then usually there's no granulomas, but I don't have any information of whether the granulomas formed normally in a CD4 or CD8 animal. I mean, we can see them by PET, but histologically, I think it's something our collaborator Joanne is doing. Yeah. So.
All right, I'm going to go with you, and then 2 online questions, and then any other questions you got to catch Patricia at the break.
Well, thanks. I was just curious. How well did they tolerate the IV injections? Like, did they get fairly sick from it, or did they tolerate it pretty well?
Of the BCG or the depletion antibodies?
Of the vaccine.
Yeah. So I was just mentioning before, they have transient effects. So CRP is up for about a week, liver enzymes, 2 weeks, but not alarmingly so. They seem like they tolerate it pretty well, but they do get, we can see that they have splenomegaly. We know from the lungs that there's an influx of cells into their lungs at, a couple months. It's hard to take down a lot of monkeys to look for this like you would in mice, but, it seems like that lasts maybe a couple months and then it resolves. But it was an issue because we've tried to pursue this clinically and the FDA was sort of not a fan of the splenomegaly. So, but that was at the high dose, right? So we might be able to bring down the dose a lot and ameliorate some of those symptoms.
I was just curious then, did you do any sort of correlation with how severe the response to the vaccine was to if they had protection as well?
No, we did. In the beginning, I did look at some PPD tests in those animals, but I mean, to your point, I think what makes this vaccine seem to some people scary is probably what makes it work, right? It's a live organism. It's persisting for a long time. It's probably replicating to some extent, and that's probably what makes it so good. So we're investigating now heat-killed vaccines, irradiated vaccines, but I'd be surprised if they worked as well as the live vaccine.
Thank you.
How did you choose the fluorochrome for the in vivo IVS staining?
I think actually there's a girl in Mario's lab named Elizabeth Potter who's done a lot of different colors. She's got some really amazing papers just on the CD45 intravascular technique. I think I just used what I thought would be brightest. I think I've used probably FITZY or Alexa 647 if I remember. It's been a little while, but if you're interested, I think this girl from Mario's lab has done a series of injections because she's done them over time on different colors, so she can look at lymphocyte trafficking over time in and out of the tissues by using like 5 different fluorophore injections.
All right, and the final question online is, I guess this may become a memory T and effector T cell story. Have you ever looked into the tissue infiltration immune cells besides the BAL?
So we have done a lot of immune characterization in the lung tissue, and it's in that first paper of the study I talked about. We did look at immune responses in lungs, spleen, lymph nodes, the skin, lymph nodes draining the lung versus distant lymph nodes, and generally wherever the bacteria go, you see responses. So there's a direct correlation between BCG CFUs being present and seeing an early response there. So related to antigen.
All right, everybody, thank you very much for a wonderful talk.
All right, everyone. So it is for our next speaker. It is my pleasure to introduce Dr. Jonni Moore. She is the founding faculty director at Penn Cytomics and a professor of pathology and laboratory medicine at Perelman School of Medicine. She has led an SRL to the NCI's highest level of recognition of exceptional for over 30 years. One of the world's leading experts in flow cytometry, she is looked to as a thought leader in cutting edge applications of deep phenotyping, flow cytometry in translational and clinical settings. Dr. Moore is also the founding director of clinical cytometry laboratory at the Hospital of University, Pennsylvania, and has led the lab until 2016. She has over 150 peer-reviewed publications and is a frequent invited speaker at national and international meetings. She holds several patents for unique applications of flow cytometry in cardiology, toxicology, and oncology, building on her experiences in basic and translational research as well as clinical diagnosis. She has led the effort at Penn to bring technologies from the newly developing field of cytomics to investigators in diverse disciplines across the campus. And personally, she's a highly respected person, but also a great mentor. She's in the past, she's never scared when you reach out to her to answer a question or give you some good advice or give you some, or be very blunt about it. And it's very appreciated. So without further ado, please, Jonni, come.
So here I am in Texas, a place where I really love to be, and here talking with cytometrists, something that I really like to do. So today I'm going to take a a little bit of approach. And frankly, I didn't know it would fit in as well as to what we were talking about today. And it's really talking about two general areas. One, how amazing and how far we've come in the technology, what we have at our hands now, and how it can ultimately benefit patients. So I'm a real bench to bedside person. That's what I want to happen. The other aspect is something that is very near and dear to me, and that is the real role that I see in the academic environment for a shared resource laboratory. I'm going to be talking about cytometry today, but it could be any, and really how the shared resource laboratory can play a major role in precision diagnostics. 21st century, we hear about team science, and it really requires a multi-level partnership. I've been around long enough where they would basically tell me, well, you really got to be the PI or you've got to be the last author. Teams don't matter so much in how you get promoted. We are now seeing something entirely different happen. It's slow to come because academia hardly knows how to reward anything but first authors or last authors. So, but the team players, and we have our computational colleagues to thank for that, because computation, biocomputation is always a team sport. But it's really become, if the goal is to bring complex technologies and where we are to the patient benefit, it really takes a real team, certainly the clinician. The clinician has challenges and problems. The basic scientists can help address those challenges or say, find out what the reasons are. And I'll get back to the resource scientist. The computational scientist works with the clinician, the basic scientist, and with us. The resource scientist is a term that we coined with some colleagues at the NIH, Mario, and I years ago, which is that we are resource labs. And there was an NIH roadmap paper that basically said a successful scientific endeavor needed parallel development of research labs and resource labs in order for that to happen. And they should proceed on a parallel path. This was around 2010 that this was designed. So we created at Penn years ago a resource scientist. It's a separate track and it's a staff level track. It's compensated at a higher level. It's a separate recognizing. So in our resource lab, we have chosen to pursue a little bit of additional path, and I'm going to talk today about how that path, how we did it, why it works, and why it makes sense. So I'm going to talk about precision medicine. You've heard about that for years. But what I'm really going to talk about are precision diagnostics. We're in an era where people understood precision medicine. You were talking about, all sorts of personalized profile, and we'll design the therapy to meet the patient. What we realized in that process, and I was involved for many years with the early stages of the cell therapy effort, and what we had a lot of tools, but we didn't know exactly how to apply those tools. In other words, going back to precision diagnostics, we had developed all sorts of new therapies. It was the heyday of pharma. More drugs were developed than possibly could be used, but we didn't have good methods to monitor patients or to identify patients that this could be successful. So this became an interest to me, and it really became possible as big data really came into the more clinical world. We had emerging technologies with new directions, new platforms, all of a sudden exploded, really probably starting in the molecular world, but expanding in other areas. We started understanding the term or believing in the term biomarkers. Biomarkers early on said there's information in the data that can be useful even if we don't know the reason. In other words, hypothesis generation versus hypothesis driven. And that's what big data a lot is. We can use big data to improve diagnostic accuracy at the individual level. Hence, precision medicine using all of these areas, in addition to demographic, can give us really precision diagnostics. Challenges, which we've heard some today, is translating these new tests into CLIA-regulated or improved type tests so they can actually get in the clinic. And we'll talk about that a little bit more. This kind of approach is really useful for unknown and rare diseases. And someone alluded to that earlier today. So the challenge is, and this is what a well-known pharmaceutical company said to me fairly recently when we were talking about that, and said during the COVID, and pretty much said, we want something that can translate to the patient in 18 months. Now, that's a challenge. And it's a challenge, and maybe some of these things happened in COVID because it was a national emergency. So things were just out there. And we tested them, and we worked on vaccines. We collaborated with John Weary and actually Drew Weissman in some of the things that we did. So one of the questions will be, how do we navigate a health emergency in the future and deploy technology towards that? And what I like to really focus my talk on there now is the Cytometry Shared Resource Lab can play a major role. And I alluded a couple times this morning, maybe the Shared Resource Lab has a new role that is in between the research SRL and the clinical lab. And there's reasons why I think the SRL is the best place for that to be. SRLs, and of course I'm biased, but are a major source of discovery, development, and translation. We provide resources and acquisition of new technology and applications. That's a lot what we do. That's our daily. We create working relationships with biotech, cytometry companies, a lot recognizing people who are here today are our partners. And we gain early access to emerging technology that becomes a two-way street where we feed back. And basically, somebody said I could be very blunt. I told some emergency technologies that they were kind of a piece of crap. And I've told some, this is good, this is promising, you've got to do this. And I've told others, boy, let me have this, I want to be right. But you have to be honest. We talk to each other. So partner with other shared labs to create technology merges. That's the one thing that I don't see as much in this field as in others, but in genomics, for a long time was a very competitive field that labs didn't necessarily cooperate at the same level that we do. And, you get further if you work together, but this community does. SRLs can provide a wide range of scientific and technical expertise. I now say that instruments in general, I've taught a lot of high school students who have worked in my lab, sort cell, and run analyzers. The instruments in general are not hard to use. What the challenge is designing the approach and mentoring people to do this that helps, that really having a knowledge to help partner with clinicians and help with people to move their projects from an idea in their head to treating their patients. And I'm going to highlight 2 or 3 of those projects that we've been involved in, one in cardiology, one in breast cancer, and one in pediatric mitochondrial disease that have come from clinicians who say, could flow cytometry answer this question? Mostly they don't know, but that's where they came to the shared resource lab and us to do it. So I think a little bit different way of thinking is shared resource labs can forge new paths, not just be service providers. Service, we've got to provide excellent service. That's the basis of that. But we can also push the edge, and we can be a critical link from bench to bedside. So through a unique set of circumstances, 2022 revealed new opportunities for cytometry as a central technology for precision diagnostics. If you said in 2019 what precision diagnostics was, the uniform answer would have been molecular testing, next-gen sequencing, those types of things. But things changed. And they changed a lot because of COVID. The genetics of COVID or any kind of genomic type testing, even strain testing, didn't provide us with the information we needed to treat patients, to look at their responses. We had to go back to other approaches. And it turned out cytometry did, because we were looking at vaccine responses. And I would, my vaccine colleagues would agree. When I see a vaccine response, it's what we make. But in a way, infection is a vaccination. So looking at those courses, and that's what we were doing a lot from when we were a central lab. We started working on those types of vaccine approaches. We closed down as a school March 17th, 2020. A week later, we're back in the lab and we're working with groups to try to figure out what's going on. And Drew was working on his mRNA and stuff since 2004. So, really doing that. So anyway, one of the things is that why is cytometry so central? So we can, in the clinical world, we're moving beyond disease definition. And I sort of complain in the clinical world that standard clinical cytometry is putting a name on something you already know what it is. That's the way clinical flow cytometry has basically approached things for 25 or 30 years, ever since we developed the lab. It's basically, and you think about, a pathologist says this is probably this, so we're going to run this panel. And then we might ask other things, or it's a limited panel. And I have hematopathologists who would tell me, well, I just want to see what the flow says, but I know it's a BALL or I know it's a CLL, but I don't really use the flow. The other thing that always bothered me is the way we analyze. We throw away most of the data. And I did research in CLL for many years. And basically what we know now, which we didn't know then, is that T cells matter in CLL because that's how the patients do, has a lot to do with T cells. We know there's changes in ratios. And actually, we published a paper years ago that showed that you could predict transformation to Richter's in CLL by never seeing B cell, only looking at T cells and cytokine pattern. So the fact is, we throw all that data away in the clinical lab. We're gating. Gating is data reduction, but it's not well-driven data reduction. So we have to change the way we think about things, and you've heard that today. We definitely are creating strong partnerships and reaching out with groups. And one of the things that we're doing at Penn, and I'll show you with our Immune Health Institute and things, is creating unique patient immune fingerprints. At Penn, very soon, much like many institutions have drawn blood where they do a bank for genomic and do genomic baseline genomic signatures. We're doing that with immune profiling. So most of those are going to be normal, and they're not going to be there for immune diseases. And our new Immune Health Institute is really focusing on that. How can the SRL directly participate? One of the ways, of course, is embracing new technology merges and understanding how things work. It takes a village. It's not all cytomics. And this is where computation comes in, because our computational colleagues can take all of the data and put it together. It's not just the one thing. We develop from the beginning, once we start doing beyond our discovery phase, we start thinking about clinically feasible protocols. Most of the really high parameter approaches are not clinically feasible. And at this point, and some of the reasons, I'll say a big thing is cost, but the other thing is that we can't do that. But that doesn't mean we shouldn't do this technology, because we need these technologies through the discovery and even through this translation phase. Apply computation, extend cytometry analysis to new domains. So I mentioned that I have some patents. I have a patent on beryllium toxicity measures in lungs. That's not a typical flow test. I have 2 patents on an early stage diagnostic for vulnerable plaque and cardiovascular disease using extracellular vesicles. Highly standardized, done, and actually had a very robust, has had clinical trials and works. I create it needs, not just me, but all of you, we need to create a very viable translational research environment. One is with biotech, pharma, other academic institutions. Again, it takes a village. The other thing that I'm not going to talk too much right now is that we have to educate clinicians on what the heck this data means. Now, immunologists and oncologists may know. I promise you, cardiologists didn't know from a flow cytometer. So telling them what this data means when they get it, the fact that we found a large number of EVs in patients who were going to have a heart attack and who had a heart attack within 30 days with a macrophage marker that's present on foam cells, that make plaque. And they understood kind of that, but they didn't understand how we look. And it was much better than NIRS IR imaging or any imaging they could do. We need to advocate with regulatory. And this is where I put on my clinical hat. Very close friend was the founding CEO of Foundation Medicine. That was the first commercial organization that did next-gen sequencing for clinical. He said the first 2 years, he spent more time at CMS explaining to them what this was. They didn't understand, particularly one thing, that why you needed any interpretation. They thought you're measuring an analyte, you give that, and physicians had the responsibility to know what to do with it. So we're creating a world with these complex technologies where we need different kind of people. We need translators, if you will. So what I thought I would do, just to show you how this all works, is tell you how we at Penn Medicine are meeting the challenge of getting new technologies to the diagnostic bedside. We've done three approaches primarily that I'm going to talk about. One is, and I put that on top because we're cytometry world here, is expanding translational expertise and services by working with Penn Cytomics and the creation of something very unique, dedicated space and personnel to support partnerships with clinicians and commercial organizations. The Penn Cytomics Translational Laboratory. I was given 3 large laboratories that are contiguous that were renovated. All of my instruments. This kind of came as we became the central lab to do the cytometry for the Parker Institute of Cancer Immunotherapy for some of the original work that we did. And we do it all. It is not a CLIA laboratory, but our laboratory director spent 15 years directing a flow cytometry laboratory at Hahnemann University Hospital, which doesn't exist anymore in Philadelphia. So she knew about the regulatory issues, how things go. I hired her when Hahnemann fell apart into our lab to run the translational lab. So she completely understands how you have to deal in the translational, in the clinical space. We did not want at this point to be CLIA certified because this is a discovery / translational lab. So we're doing other things, but we did everything in the same approach, the same documentation approach, very standardized. I've talked to you some, we created software. We have a 6-person computational group embedded in our group. 2 of them are biocomputational physicists. The other 4 are programmers. They have developed specific software under guidelines, regulatory type guidelines, that will help us do what we want to do. So we've created an environment where investigators, clinicians can bring us a clinical question. And it's at different levels of development. Some already have a test and they just want to get it to a point where they can translate. Others are like in our cardiology world, my cardiology colleague, who is the director of vascular medicine at Penn, came to me and said, if a patient comes to me and says, "Doc, am I going to have a heart attack?" There is no test I can do to answer that question. If he's going to have a heart attack in a few days, his troponin levels might be high, and I can say that, but I don't know. Genomics patterns have never proven to be 100%. They're involved, but we all know people who had bad genomics, bad lifestyle, who live long, and the opposite is true. He said, so I don't have any. He said, can this flow cytometry stuff help out with this? So to his surprise, I taught him well about this, and he helped to develop our test. Then, but the point was, he came as a clinician with a project. Secondly, we've developed at Penn something very new in worlds of pathology. There's a new division in our Department of Pathology and Laboratory Medicine. It's called the Division of Diagnostic Innovation. And this is where we have several bioengineers that are being hired into this, clinicians, computational people, bringing people in who can focus on new technologies with the goal to get to that lab, get to the clinical lab. This just started a little over a year ago. The department of school put a lot of money into it for recruitment of people. And I will mention just briefly, one project that we're looking at. I'll put a plug if you want to hear more about that project, which is interesting. Come to Montreal. The speaker is going to be there, and she also won last year the Presidential Award for the best oral presentation. So it's very good. We wanted to, I mentioned to you the immune profile, which is the prototype of the Immune Health Initiative. Penn has initially put 25 million in this program, building a brand new building separate from that and going to put more into it. And that is where a guided, focused, rapid development to patient pipeline of really mostly immune-based diseases of some sort, but using technology like cytometry. We are the cytometry partners, mass cytometry, imaging, immune assays, begin with the goal of doing this quickly. So together, these initiatives, I think, are going somewhere. So what did we do? A bit about the translational laboratory. So you might say, and I get this question, well, this was done within the SRL. How do you have the time with your personnel and all to do this? This is a fee for service, this whole thing. So yes, it is. We pay people. We get the right group of people. Our lab is in there, and it uses technology that we already have staff to do. So it doesn't take away at all to what we do. We've hired like 3 or 4 more people to do this so we can turn this over. Simply that we look on a variety, we choose what we're going to do. So we translate promising discoveries to clinical utility. Like if it's really early stage, we might suggest to the investigator, we still might be involved in it, but it may not be involved in the translational lab. It may be, well, you're not quite there yet. So a couple of things that we really think are important. Of course, to get this process going, it has to be all SOP-driven processes. Coming from the clinical lab, well, I know that. Coming from the research world, I can't tell you, and I'm sure all of you have seen this, how many times I have people come and they have a problem, and I say, well, what's your, what did you do here? Well, I used to use this kind of approach, but I changed it when I was doing this, and now it's different. So it's not standardized all the time. That is very important if we're going to use computer-driven analysis, because like my computer people say, you give us data, we'll model it, we'll probably find something important. But they don't, garbage in, garbage out. It may not be garbage in, but it may not be the data that will address the question you're asking. The goal for most of these tests is to rapidly translate them using the LDT pathway, because that has in some ways been loosened up a little to allow tests to get to the clinic more than it used to be, but using CLIA processes. And ultimately, we don't want to be the clinical lab. We want to create something that can be handed off. Now, do I think most clinical labs are yet able to accept that? And we heard from David this morning. There's got to be a parallel evolution of the standard clinical lab. But that's where it should be. It shouldn't be in a research lab, and we need to develop processes, regulatory and qualifications and all to allow this technology to go into the lab. It's not going to be a 37-color spectral panel. That won't see the light of day for a while. That's his phase 3 in some ways. But why won't it see it? Because cost drives hospital clinical labs. Hospitals have a hard time justifying 10-color diagnostics. And it's just, we're not in the world yet where we can pay for 37 colors. So Clinical trials, yes. But clinical trials will serve to tell us. So one of the goals is to try to take the results of clinical trials and can we package this so it can go to the lab? Does it mean it won't be done? Not necessarily, but it's not going to be available to everybody until we can do that. Educate clinicians, as I mentioned before. And very strongly, I can't emphasize this anymore. One of the strengths of our field, as we have had close commercial partnerships to develop this field. We wouldn't have the tools we have today if the companies didn't have the relationship with the users. And I find this, and it's different because my husband is a clinician and he says, oh, you mean it's like drug companies? We never pay any attention much, not like drug companies. It's very different. We are very interdependent, if you will. We have to drive that, and that is very important for us to maintain. So just to start closing this out in a way, I'm going to tell you about our pipeline, which is three areas. Our most advanced one is an assay that we developed with our colleagues in cancer oncology. It's a minimal residual disease, essentially, assessment for breast cancer. It is a detecting in patients who have been treated for primary breast cancer. It's been known quite some time that there are surviving cells. There was an assay developed some years ago. It was a cell search assay, which looks for circulating tumor cells. Problem with that assay, it's not that sensitive and it's definitely not that specific. So we said, well, can we use the approach? Data suggested that the place that these disseminated tumor cells, as they're called, hide out is in the bone marrow. So if we could find a way that could two applications, one to assess the efficacy of initial therapy. Did we get as much as we should? The other is to monitor patients for a period of time over a while. Yes, it has to be a bone marrow aspirate, but believe me, we're not having any compliance issues because so that we detect the re-expression way earlier than it can be detected. This is important because right now, with the level that we can detect, relapsed breast cancer has a very high mortality rate. So it's very important. So we decided to do that. So we did it, and you're going to appreciate this because it includes working with several different vendors and even creating some issues. We're now on our second large-scale clinical trial. The first one had 5,000 patients. This one's going to have 10,000. So that's good. So we call it DTC flow liquid biopsy. We basically, patients who are enrolled in the trial, the DTC flow, we get bone marrow from them. We separate those cells, and we've done it 2 ways. Initially, we did it using an instrument or an instrument add-on that BD had called the BD Fax Focus. The downside of that was BD decided that it wasn't worth their while to continue to make that. So we could do it, but no one else would ever be able to do it. But what that did basically was it was an add-on to an influx that basically used acoustic focusing to focus cells, but that was after antibody depletion, magnetic antibody depletion. So it was really an enrichment technology that did not involve really any washing, which is very important because these cells are at a frequency of 1 in 15 million. So we needed to do that. So then we would enrich those cells, run it through in our initial process. We gained it on CD45 negative cells. And we used standard approaches. We looked at EpCAM, which is what Cell Search did. We have now expanded to a 10 marker panel so we can simultaneously assess ER and PR, HER2/neu, whole variety of markers that we know. So we're way more specific than it was. So with this approach, and we collect a whole lot of cells, like I said, at least 15 million, but we can do that with this enriched population. Before, the population was so tiny, it would take hours. Kind of reminds me of the olden days when we were collecting in the first cell therapy work, suppressor T cells. They were very rare, and it would take hours and hours and hours. And that's what the FACS focus was developed to address. Once we got that, it took us 2 hours to do it. Well, this is the same idea. We would take those cells, and we did, you'll see an image stream point. We did staining, looked at DTC markers, and we actually to see that what was really staining. And we also did downstream molecular analysis to show, and we were able to show that these re-emerging cells have the same genomic abnormality that the original tumor had. So we knew we were collecting that. We were able to detect a HER2 low population in breast cancer in a patient with many patients now with triple negative recurrent tumor. And we added single cell genomics again for enhanced detection, both with prognostic and therapeutic significance. And you kind of can see some of these, there's some plots there that say and sort of show you how these populations we were able to detect. And over on the right, you can see how we correlate with some markers. So how do we do this? Well, I told you we had the FACS focus, and the first part of it was, like I said, magnetic depletion of unwanted cells, inline acoustic wash. We had pre-stain, we did index sorting, and then flow cytometry analysis of cell surface proteins. And then we did whole single cell of that. That's how we proved this worked. The problem came, and worked really well. The problem came when PD not only stopped making this, but all the engineers who were making it left. And now, however, one of those engineers decided to start her own company to make essentially this. BD was okay with it. They said we weren't going to do anything. So that's this. Before, we were doing it with an add-on to the sorter. Now, this is a freestanding unit, can be used with any sorter. It's made by a company called Applied Cells, and they're very new, but they won, I think, the innovation award at Cyto 2 years ago, maybe 1 year ago. And they have made this, which will have multiple uses. It does the same thing, but it does it to make it used for anything. So now we have a couple of those, and we run 2 or 3 patients a day, and we're able to do the trial. I'm not going to talk a lot about all our EVs. Extracellular vesicles have been an interest of mine for a long time, but we're doing a lot in that area. I mentioned what we found, but so I'll skip that part over. In the process, we developed a computational platform that we call Cloud Cytomics. and it enables accurate and sensitive EV analysis. There's a lot of software in that. We use it for ourselves. It's never been commercialized, but we actually let some people use it. So we're there. Last couple of things I mentioned is I talked about the Parker Institute. So the first application of translational cytometry was with the PICE model partnership. We did all the things that I've mentioned to you. We worked for 3 years. There was a trial called the Prince trial, which is a new immunotherapy trial. And we actually did all the analysis for all the samples. In the end, what we found was we identified biomarkers, pretty much a single biomarker, in 2 arms of the trial that using a 28 color conventional panel and biocomputational approach, that the marker we identified for each one was highly predictive of response to therapy. This is just this, I'll let you see, but this was the paper that was published in Nature Medicine in June, and it was the first time a high parameter approach had been used in any clinical trial that had definitive value. There were other technologies used, so that really was our first thing. So basically, the last thing I want to mention is our new thing with mitochondrial disease. And basically what happens here is children who have mitochondrial disease don't get cancer. So we're looking at this with a group at CHOP to try to develop mechanisms. This is very interesting, and this is where this is very important. If I tell you this very classical genetics group does not know from immunology a T cell from a B cell, this is absolutely true. They just don't know immunology. And let's just say in the process we've been looking, and I would have in my mind thought that of course you look at immunologic markers in these 2 areas. We're finding something there. This is the 37 color full spectrum panel that we're using. The markers of primary purposes on here, and I can talk to you about later, but it does a broad immune snapshot, kind of like we're going to do with the fingerprint pattern. This is what we're looking at. We use this to define the fingerprint of those patients. And we're going to, our next step is going to be looking at mechanisms. And we have a panel design, we're ready to go, that looks at apoptosis in all its forms, both in autophagy and everything. We're looking in these cells. So finally, I'll mention, I talked about the division of diagnostic innovation, which gives us a structure in the department. This is one example of what people are doing, what an investigator is doing there, developing a new EV droplet technology and the instrument to measure it, and she's been able to combine with genomics. So really having people who are working in that area. But the final thing is these new genomic and proteomic approaches form the basis of immune profiling. We're doing this in patients, and we've learned a lot of lessons to sum up. We're going to create a separate space for translational SRL. We've done that. Engage clinical partners. Be well-versed in regulatory and procedural. Tests must be rapid, reliable, and precise. And no one test domain will be enough. And the others we've talked about. So my last slide is really the bridge from research to the patient is enabled by ideas and expertise from SRLs and resource scientists. We created the Penn Cytomics Translational Laboratory to be a prototype laboratory for this type of work. The path across the bridge requires a variety of things we've talked about, but in the end, you could take your SRL to the next level by becoming a research clinical bridge. Together with academicians, clinicians, and biotech, we can revolutionize the world of clinical diagnostics. We have the tools, we have the knowledge. So I would say the stars have aligned. Cytometry is poised for a new birth in precision diagnostics, and the SRL is front and center. Thank you. Yep.
A little bit into the break. If we can take, do we have any online questions? Okay, let's take Dan. Would you have a question for Jonni?
Thank you, Jonni. Big data often means big algorithms. So we take our flow data and we push it through UMAP embeddings and clusters and neural networks. How do you take the output of these algorithms and bring it back to something that a clinician can actually use?
So the algorithm, so this is the thing, when you take big data, sorry guys online. Basically, that's still in your discovery phase. One of the big issues is to translate that to information that clinicians can use. And that's something that it takes partnering with clinicians. We're not going to give them the whole process. We're going to give them something, it may be visual. They like visual. So it may be that, but we work very closely with clinicians to say, we usually find somebody who kind of has a clue on biocomputation, say, well, how do we need to do this so that you can understand? And we did a very visual for the cardiology one because they really didn't understand. We're going to have to do that for the mitochondrial disease, I think. But that's what we do. You get the data, and then the final translation step is very important to create something that the end user can understand.
Thank you.
Thank you so much. All right. Thank you, everyone. Now it is time for a coffee break. We're going to take a 20-minute break and be back here in 20 minutes. And we'll start in exactly 22 minutes to be precise.
introduce. So today's my lucky day because I have the pleasure to introduce amazing speakers. Now I want to introduce our next speaker, Patricia Rogers, who is currently the Associate Director of Flow Cytometry Facility at the Broad Institute of MIT and Harvard, and has worked in the field of flow cytometry for over 16 years. Tricia and I actually have been friends for many years, and I've had the pleasure to work and collaborate with her many times. She's one of my biggest inspirations. I say this a lot, on how to build a successful core and a successful career. From her humble origins to starting as a technician at Harvard and ending up managing the whole core facility, and then being invited to create and lead the flow cytometry platform at the Broad, which I consider one of the most successful core facilities in the world, and well managed. Her core supports around 400 researchers from over 30 groups and programs. The facility has grown rapidly and now includes over 20 flow cytometers, genomics, and imaging instrumentation. Tricia was the first person I know to have had the vision to merge flow cytometry services with single-cell sequencing technologies, while most of us were still trying to figure out why most labs were asking us to sort single cells onto plates. She's an inspiration to every woman who struggles to build a successful career, but most importantly, she's an inspiration to all of us who aim to build world-class facilities. So her talk today, What's Next in Flow Cytometry, is actually a segue of her talk last year here at FlowTex, where she discussed her experience in merging flow and sequencing services. So Tricia, thank you again for accepting our invitation, and welcome again to FlowTex.
I'm like blushing, like, thank you, Rui. Always like hype me up so much. Well, thank you for the program committee for welcoming back this year. I had so much fun here last year. And actually yesterday I went out and bought some cowboy boots. So if you invite me back next year, I'll buy a hat maybe, and then I'll just keep acquiring more Texan wear, and I'll just be officially Texan at some point. So yeah, as Rui said, I talked last year here at FlowTex about expanding your flow cytometry core to include more genomics, which I think a lot of us are kind of dabbling in right now. And this year I want to talk about where I see the future of flow cytometry core facilities going. And obviously this is my opinion and what I'm doing now in my own core facility. And this might not be happening for a while, and I don't think we're going to be out of business. There's still going to be a need for flow cytometry, so don't worry. But I just want to share some of my experiences over the past year since I last talked to you, and also kind of the direction of going in. So my talk today it seems maybe a little disjointed, but all of these kind of are going to be connected by the end of my talk. So how I expanded in the past year, what is the new technology, what's like the latest and greatest, and how that is going to shape our core facilities. What are some pain points that we're all experiencing in flow cores? And then how we can learn about these new technologies, kind of where can you go to get this information? Because it just won't fall in your lap. So let's start about going into detail how we expanded last year. Maybe just to highlight, if you haven't heard of the Broad Institute, we're a nonprofit, kind of a private research institute that's affiliated with MIT and Harvard. We're right across the street from MIT in Kendall Square. So across the street is the Koch Cancer Institute. And we're kind of divided up into 3 areas. We have scientific programs, we have our platforms, which a lot of you have heard of, especially the genomics platform, where you do tons of sequencing, and started actually out of the Whitehead Institute as the Human Genome Project. And then we have our little recharge centers within the institute that support the platforms and the scientific programs. But mostly we focus on human disease and you know, how we can solve these problems that we're all plagued with, you know, cancer, cardiovascular, diabetes. We just started an infectious disease and microbiome program, but we also have things like kidney and obesity. And then we got a giant donation from the Stanley Center to start a psychiatric disease research area. And we also focus on rare diseases. So a lot of these diseases that there might be only 200 or 300 patients in the country. So we're looking at cures for that as well. And so I fall in this recharge center area, and I'm the only core facility at Broad, technically. The platforms are really like collaborators with the scientific programs. So the imaging platform you might be aware of, Ann Carpenter runs that, and that's only software. Metabolomics and proteomics, Steve Carr runs that, and he does a lot of, he's really about publishing papers and working with the scientific programs. And so he doesn't want to run the day-to-day samples that I might run. And data sciences, they've done a really great job at creating GATK, which is a platform that actually Illumina uses on their new sequencers. And so we have all of these really like well-known platforms, but at the end of it, they don't want people coming in and messing with their stuff, or, they don't want to train people how to do things. They're really these like big collaboration powerhouses. And that's really our strengths at Broad, is being able to do these huge projects that other places can't do. And so last year I showed you this slide in my talk about what we had in our core facility. So there's a lot here, and I kind of broke it up into flow cytometry. We have a lot of sample prep because that really feeds into flow cytometry, molecular biology. So this is my genomics. So we're really a 10x genomics heavy lab, and then some imaging, very light on imaging, but I'll show you more of what we're getting into in that area. And then just in the last year, this is what I've added on the things that were already on this list. So every year I kind of add another analyzer, another sorter. So the things in green are what I've added. The one instrument in red, we kind of discontinued that because Bio-Rad's discontinuing it. So we didn't really want it taking up space because we are very constrained with space. That's our biggest lack of resource. But we're getting a new building in 2025. So I'm already staking out some lab space because we'll need it. So yeah, we added another sequencer, we added another Chromium Connect. So we are really growing at a pretty rapid clip. And then last year I told you, these are the things I wanted to add in the upcoming year. And I actually did add all of these things. So we added, the Xenium is getting installed as I'm here right now in my lab. So that's a spatial transcriptomics platform. We added more Visium services with the CytAssist. We have added also a Luna4 Comet, which is an instrument I'll talk a little bit about in a minute. And, we're expanding into the imaging area as well because, our strengths as flow cytometry cores are, I used this word with Kelly yesterday, democratizing access to easy to use equipment, right? That's really one of our strengths. And it's really been progressing over the years that vendors are releasing easy to use equipment. Other technologies are also doing the same thing. And so finally, Leica came out with a very easy to use confocal system called the Mica. And we added this to our core in the last year, and it's been really great. It's a 4-color system, can do confocal and wide field, and people love it because it has an incubator built in. You don't have to move a box and put it on and then take it off. It's just all in one kind of system, and it's very easy to run and operate. We've added also laser capture microscopes because we do a lot of single cell, and we're getting into single cell proteomics, and so this was really important. And just histology has been a big thing that we've invested in the past year. And you'll see why in a minute, but it's just nonstop. So it's a lot of fun learning new things, but it can also be a little stressful. And so in the upcoming year, maybe when I come back next year, I can tell you about all the other things that we've been adding. But not that I'm like saying you have to invite me next year, you don't, but if you do, spatial transcriptomics and protein services, there's so many vendors coming out this year with new equipment in this area. It's becoming a very busy segment in the technology world. So there's Resolve and Luna4 and 10x and NanoString, and you can just go on and on. There are a lot of companies in this area, and they all want your business. And so which one should you buy? We don't know. So we have most of these technologies at Broad. We're testing them right now. Hopefully there'll be a paper out in the next year that will say this one's good for this and this one's good for that. So we're doing kind of just a standard bake off at Broad right now. Investing in more histology. That's going to be really important because spatial relies on histology, and a lot of histology cores are at hospitals. These cores are very heavily used. Obviously, they're having to process all of these samples for patients. They don't really process samples in a way that might preserve RNA. And so you're having to work at their whim, right? Of how they do things. And so having this equipment in a research core setting allows you to tweak settings and find the best method for processing your tissues, from perfusing them with different chemicals to working on your staining protocols, all of this stuff you can then tweak in-house and make it the best that you can for your spatial transcriptomics work. And then we obviously know that this past year, image-based sorters have been coming out, BD with their Discover, but there are also other companies, DeepCell and ThinkSight. So ThinkSight, if you heard of the Ghost Cytometry paper from Japan and the University of Tokyo, so that is also very exciting. And I'm hoping we at least get 2 or 3 of these instruments in our lab. And then easy to use and cheaper sequencing. So Illumina has some competition finally. It's just not Illumina PacBio now in the sequencing space. There's Element and Singular. So we're definitely looking at Singular right now because it seems to be a good segue from 10x to Singular and very cheap, almost $200 a sample to sequence for 10x. So if you haven't heard of them, I would definitely check them out. So let's talk about some of this new technology that I just threw at you. Well, first, like, what are we measuring in single cells? So not only do we measure genomes, but we measure transcriptomes, which is a lot of what you guys do when you sort your single drops into plates. So that's a single cell RNA-seq. We do a lot of epigenome work where we're looking at histone modifications in cells, and we do this at the single cell level now too, but also proteomes and now metabolomes. So that is becoming the next big thing that we want to measure at the single cell level, which is pretty impressive if you think about it, right? That we have all of this stuff in one cell that we can then get all of this information out of that one cell. So why do we care in a flow core? Well, we have a lot of competition, right? So there's a lot of different instruments that can do the same thing. So in my lab, I have a Sony ID7000. I just installed the Luna 4 Comet. This is a company out of Switzerland. And we also do 10x work, so we can do CytSeq. So, which one do you choose to measure protein expression? You have a lot of options here. And, if you're working with blood, then it might be, okay, well, I have to use the Sony. I can't do, tissue-based analysis using the Comet. But what if you have tissue? You don't have to dissociate now and run it on a flow cytometer. We can now analyze this other ways. Or if we do dissociate it and we're going to do single cell RNA-seq, we could just do CytSeq in combination with that. And so there's a give and take here because some of these processes might be harder to optimize or they might be more expensive. CytSeq is still pretty expensive if you think about it on a per sample level. The Comet can only do max 40 parameters. And so if you have more than that, then you might not want to use that. And also if your tissue is something that's hard to section, or sometimes we work with bone biopsies and those are a little bit harder to put on slides. So there's just a lot of like, what do I do kind of situations, right? And so I think as a flow core, we're really set up to talk with people, bring them in, and as Anna was saying in her previous talk, really have a service set up where you can sit and discuss all of the different options and how you can help them. And that's really what we're doing in our core facility now, is that we're bringing people in, we're having consultations with them, and we're picking the right technology for them to go and do their experiments with. And maybe they do a little bit of everything too. And that's the nice thing about having everything in one space, right? You can go from one instrument to another until you find that, like, the Goldilocks scenario, the right one, the perfect fit. And so really what I see the future of flow cores going to is this technology sandbox where we have a little bit of genomics. We're not going to compete with our genomics cores because, I'm not going to get a NovaSeq X, that instrument's $1.25 million, and it does, billions and billions of, I don't need that. What I have in my lab is, little MySeqs, and I have the NextSeq 2000. So I can do some basic stuff, right? And then I have all of the variety of flow cytometry instruments, and I'm getting into imaging with my slide scanner and my Mika. And then I have my whole little histology setup. So what's the nice thing about this is that a researcher can come in and kind of go from start to finish to every technology and do proof of concept experiments. And then once they have something kind of established, they can then send that off to their larger core, so the genomics core, which they can do high throughput work for them, or the imaging core that has these really nice, high throughput confocal instruments, maybe the Phoenix Opera. And so we're really just giving access to everyone to do their own kind of tech dev. And so that's where I really see the strengths of flow cytometry cores, and where I see flow cytometry going in the future, is where you can have a little bit of everything in one space, because we have the skill set to help train people and to sit down and talk with them to get them going. And so I just want to highlight a little bit more about spatial transcriptomics, because I think this is a really useful tool to bring into a core facility because these instruments are becoming easier to use. And I just grabbed this little graphic from Wikipedia because I thought it was a really nice illustration of just the basics of spatial transcriptomics, because we had a town hall recently at Broad on just spatial work, and the slide, it was so messy because there's so many different assays out there right now for spatial. So there's a lot of homebrew coming out of individual labs. There's a lot of companies coming out with the products from these little labs. So it's just all over the place. But you basically have, 5 different ways that you can do spatial. And I don't know if microdissection is really considered spatial or not, but this is a way that you can isolate single cells and then do single cell sequencing on them. But if you start with in situ hybridization, this is things like fish, merfish, RNAscope, and, commercial products now available are like VizGen. And so they really are able to go in with probes, bind to the mRNA, and then image that using usually either you can have your own fancy microscope or you can have it built into the system that you bought. So 10x Genomics just released their Xenium, which is also similar to this. And that's what we just installed in our lab, because it is a pretty easy and straightforward system to use. So it's very easy to put your tissue on the slides. You put all of your reagents on the instrument, your slide on the instrument, and in about 3 days, you get all of the data out. Now that, all of that data is going to be a lot of information. And so how you transfer that around is going to be a problem, but I'm sure we can figure that out. In situ sequencing, I don't know if there are a lot of instruments out based on this. So 10x Genomics actually bought the company that had this. It was ReadCore and Cartana, and that's kind of what the Xenium has come out of. But they didn't include this in situ sequencing where you actually sequence inside of a cell, which is crazy. So that application is called FISSEQ. So it's like FISSEQ. But I don't think anyone's actually commercially releasing this yet. But it uses this padlock-based amplification inside of cells, and it's pretty cool. So we'll probably see that in a few years, though. And then we have in situ capture, which is a lot of what we're doing too with 10x Visium, or Curio now has commercialized SlideSeq, where you have these arrays on slides. So you have the oligos already placed on the slides, and it's like little dot arrays, and then you put your tissue on top of it. And then you permeabilize your tissue, and then those probes will bind to your tissue, the mRNA in your tissue. And then you take an image of that, you get it off the slide, and then you do everything like you would for normal library construction, and then you sequence it. So you have either probe-based spatial transcriptomics, or you have sequencing-based spatial transcriptomics. And, they have their pros and cons and their strengths and weaknesses. But we do a lot of this in situ capture in our lab right now. We will be doing the in situ hybridization soon. And then the last one is just in silico, which means they're just taking a lot of the data that's already out there and kind of reconstructing. And so we don't really help out with that. So we mainly do in the probe-based or the transcriptomic-based in our lab. So this is kind of again, from the Wikipedia spatial page. I thought it was a nice little diagram of just how the Visium or SlideSeq works, where you have all of these arrays on your slide. The only drawback to this is that once you put that tissue on there, you can only lift it up one time and reposition it, or after that, it's kind of stuck to that slide, and these slides are, many thousands of dollars. And so if you mess up, you just wasted a lot of your PI's money, and so they might get angry at you. And so this was one of the things with doing Visium that researchers were really kind of antsy about doing this themselves. And so having us stand there with them and walk them through the process has been really helpful for them. And so us offering this as a service has been really useful as the flow core. Also, because you can charge more for doing this. People tend to pay more to do genomic services than they would for flow cytometry services. But the resolution of this was not so great. They're coming out with a higher resolution, because it's not single cell. So you're getting a cluster of cells at each spot. But in the next year, we should have near single cell resolution through the 10x Genomics. And I think SlideSeq has a much higher resolution than the 10x right now. But 10x did release this nice little instrument called the CytAssist, and we implemented this over the past year in our lab, and we really love this because now this makes it very easy to put your tissue on a slide. Instead of having to use those really expensive gene expression slides and make sure you get your tissue in the right area, you can just now put your tissues on regular standard glass slides, do your H&E staining, and then use this little instrument where you put your tissue on the glass slide, and then you put your gene expression slide, and then you, over here, you sandwich them together. So it just transfers the tissue from your regular slide to your gene expression slide for you. And then you do all the work on this little instrument, and it makes it so much more streamlined and easy to train on. So this is for the probe-based. You can't do this for the transcriptomic, like the 3' transcriptomic or 5'. So this was, you have your mixed set of probes. So you'll have your 20,000 probes for human, for example, and you just put that in there. And so that's why you don't need to have, you can use this instead of the gene expression slide with the probes already on the slide, because you're adding the probes on top. So then you go through, you do the same sort of library construction and probe extension. You don't have to do as much hands-on work here at this point, because you're not doing the standard library construction with like the extension and ligation. You're just adding your index primers before sequencing. So if you want to get into this, I feel like this is a very good area to get into it, is by buying this little instrument to help people with doing Visium. Not that I'm trying to like sell more instruments for 10x, but it's a very easy instrument to use and to train researchers on. So if this is like an area that you've been trying to get into and you're not sure, this is like the area where I would just, right now it's for FFPE, but they just launched a fresh frozen as well. So, and I can obviously, if you want to talk more about this, I'm happy to. And then I'm going to finish today talking about the pain points we experience in flow cores, which is kind of related to these areas, which I think all of us have had issues with hiring, finding funding to hire people, and how to keep good employees. Rui just said he poached Anna, right? And so that happens a lot where we poach from each other. But how do you say, hey, you should stay here and not leave my lab? And some of the things I showed you before, so all of the genomics work, that can also help to lead to funding to hire more people. So finding future employees, I haven't had too much of an issue with this myself. I don't know if it's because I'm at Broad and people want to work there, or because they like me. I'm not really sure, but this is a great way to find people, is to network with other flow cytometry groups. And so I actually hired somebody via FlowTex last year who's starting in my lab in a few months. So going to other outside of your network, maybe, there's New England Cyto, there's the Southeastern Flow Group, SEPSIG, there's this one. Some people might want to move around. And so I think that is a great way to find new people. And Rui did it too, right? He hired Anna. And then whenever I have a job posting on my website open, I always have an open mind when I'm looking at non-typical applicants too. So somebody might apply to my job role for a junior technician role, and they have no flow cytometry experience whatsoever, but I see that they have good customer service or something like that. So one of the people I hired, she worked at CVS at a pharmacy, right? Well, that is like the most stressful job I can imagine somebody can have, is working in a pharmacy as a technician. And I said, you can handle people, no problem. You can handle researchers all day long. And she can. She has a very level head on her. We did hire her. She's been really great. She's eager to learn new things. And actually the histology side has been right up her alley. And so she's learning all of the new histology equipment and training people how to do all of that work. So that has been awesome. I was really happy to hire her. And then at ABRF this year, we're going to have an entire workshop focused on creating curricula to work with community colleges. And this is going to start with just flow cytometry. So the flow cytometry research group has been meeting over the past year to write this curricula, to then reach out to community colleges to do apprenticeship type work in flow course, which I think is an amazing idea. And I think we need to do more of that. So I don't know if you're planning to go to ABRF this year, if any of you are, but it is virtual, I think. Maybe the workshops aren't, but if you can attend virtually, I would highly suggest it. There are going to be breakouts for some of these things too. But I think they're gonna continue on in this strain. So they're gonna keep going forward with the community colleges. They are actually inviting community college members to come to ABRF for the day. So we're having a session for them to come out and learn. So I'm really excited about this, because I think this is really going to be the way for us to find future employees in flow cores and just technologists in general. And ways to find funding. So that is one of my limiting factors, is that, I have to prove that I can support my FTE before I can hire them. And sometimes it's like the chicken and the egg, like I will have future work that will support this, but I can't do that work unless I have this person in the lab, right? And so I really battle with this. But some of the ways that I've been working around this is collaborating with PIs to write grants to fund employees. Sometimes they have grant money just sitting there, and they can actually, it's easier for them to partially pay for your employees than to pay you hourly rates sometimes. And so you can say, hey, can you cover 5%, 10% of my employee? And then I'm not going to charge you to use this instrument, right? And so you can kind of work out these side deals that will just bring your overall budget down a little bit, which makes it easier to then justify hiring more people. So those are some of the creative ways I've been trying to find in order to support my hires. And then also bringing in instruments that can subsidize hiring. So if you have an instrument that can run 24 hours a day and collect revenue, then that can support, hiring people. And so sequencers, for example, they run 12, 24, 48 hours at a time. And so I'm collecting revenue on every hour that they're running, and I have 5 of them in my lab. And so this is like the third, after the Cytoflex and the Sony, this is my other highest moneymaker in my lab. So I think, I don't want you to go out and step on your genomic core's toes, but if you work with them maybe and say, hey, is there a way that I can do this as a service and not take away your business? Some of them don't have MySeqs anymore because, they're not really worthwhile for a big genomics core to have that. So there's just creative ways to, find extra revenue. And then retaining employees. And so this is something else. I talked about all of this new stuff that we're adding to the lab. Well, people really like to learn new things, it turns out. Like they like to have, engagement in the facility and not to be doing GFP sorts every day, right? Like we like to learn new stuff. And so, bringing in that new technology has allowed me to retain my staff, although Rui did poach someone for me too. So, I mean, Rui's just like taking people left and right, but she was moving to New York anyway. So that's what I tell myself. But sending employees to as many learning opportunities as possible and telling them to engage in these, societies. So, I've really told one of my employees to engage in the Cyto Women or the National Society for Histotechnicians. And so we're really trying to connect and collaborate and learn. And so, and also if you can convince your institute to allow you to pay your employees just a little bit more than your standard RAs, that goes a long way, because I don't think they can see the difference between, an RA working in a regular lab who might be there for 2 to 3 years and goes off to grad school, and an RA that joins your lab. There's a big difference because I'm teaching my RA to teach other people. And so if you think about it as, people who go to school to learn how to teach, you usually have to get a master's degree to do that, right? And so, it's a very big difference. And so we should be able to pay our starting RAs a little bit more, even if they are coming in with low experience, because we want them to stick around. I don't want them to leave after 2 to 3 years. So those are just some of my takeaways from retaining my employees, because I do have a pretty good record with retaining workers in my lab. And, if I had the choice over sending somebody to a conference or me going, if I only had the money for one, I would probably send my employee rather than me, because I already know a lot. I've had that opportunity, right? And so I really want my employees to learn as much as they can, and so they can grow. And so where can you go to learn about new technologies? I did mention the ABRF. So that this year it's happening in Boston. And so it's before Cyto. So if you can get away and go to that, I would highly suggest it, because it's all of the technologies. And this is the Association of Biomolecular Resource Facilities. It started off as genomics, but it's metabolomics and proteomics, it's imaging. We've just added histology this year. And so we're really trying to collaborate with all the technologies. And so we can all learn from each other. So there will be, flows, cytometry focused breakouts amongst all the other technologies as well. There's also a really interesting breakout on shared instrumentation grants. This is the 40th year anniversary of that program. And so we're going to have a plenary and a breakout. I'm on the program committee, if you didn't know. That's why I'm plugging it here. The National Society for Histotechnicians is also in Baltimore this year. I sent 2 of my staff there last year, and they actually were able to learn a lot and connect, because this is a more hands-on conference. And so they do things like workshops where you actually have to go and do work. So it's a little bit different than Cyto and ABRF. And so I would highly suggest this if you're thinking about adding anything histology related to your core. And then Advances in Genome Biotechnology Conference is always a great one if you want to get more into genomics. This happens in February in Florida for the general meeting. This one gets filled very quickly. So as soon as they open registration, if you want to go, register right away. But this is where all of the new technologies, this is kind of like the highlight for all of these vendors. This is the big release for all and launches for all their products. If you are more interested in precision medicine, then they have their fall meeting, which is not in Florida, it's somewhere else. I think it's in September though. But I usually go to the one in February, the general meeting. So I just want to thank my team. We're small but mighty. We do a lot of stuff, and there's only this many of us. And so I have AJ and Andrew, who are the techs, Benoit, who's on top of his boat. He does professional sailing in his free time. Jen is our wonderful admin who does all the paperwork for me, so I don't have to think about it. And then Natan is the lab manager, and he is a jack of all trades. So I would be lost. If you poach him, Rui, I don't know what I will do. Yeah, you better take note. And this is my house 1 week ago. So for size reference, this is a collie and a corgi. So the weather is much better here today. So, and I guess we're going to end there and do a roundtable. So thank you very much.
I'll invite the speakers to sit here in the not so round table.
Are we waiting for questions?
Yes, no, so we're just trying to
get you on camera.
Put the camera.
Yeah, I don't know how to do it.
Me neither. Yeah. So
I
cue
know
camera.
I know I've been told.
So, so what we'll do is I'll basically ask questions. So we have some questions from our online audience, but we'll start here with our in-person audience. And I guess if you have specific questions for one of the speakers, just mention. But I would say that all of the speakers can, add their opinions to any of the questions. So, so I don't know. Do you guys have any questions? Yeah, go ahead.
Okay, my question is specifically for Anna about your backbone panel. I know that you guys have auroras in the core. Are you going to test it on any other spectral instruments, or are you also going to test it like on the Fortessa or any other instruments to see where else it can work?
Thank you for work. Thank you for is it? Thank you for asking the question because I forgot to mention the. So yes, we are testing different spectral instruments because the idea is that the backbone could be used for any lab that doesn't specifically have the aurora, right? So we have the S6 spectral. We already run the backbone there. And we are gonna have the ID 7000 soon. So we installed last week, right? Mm-hmm. So the idea is to test in the different platforms to see if we get the same type of resolution or if there's something that we should be more concerned about using one or other instrument, but just the time that we can publish this, then if you don't have the Aurora, you can use the other one. Or so it's not a specific for one instrument, but the idea is to keep for spectral flow cytometers. We didn't have we're not concerned like, okay, let's not put these and these for, because maybe for conventional. So the idea is for spectral, but maybe we can test as well.
And then just a follow-up question to that. Are you going to look at, autofluorescence and maybe indicate whether you have to use it with the tumor, but not with spleen and so on and so forth?
Yeah, it's a really great question, because I've been spending a lot of time trying to search different autofluorescence for at least these 4 different tissues that we are acquiring. And of course, always improve a little bit the resolution, especially because we are dealing with a bunch of different myeloid cells that they have different autofluorescence. So it's helping a lot, playing a little bit with the AF. So the idea is I want to conclude in the end of the paper that how useful it is, and if you need to have different approach for different type of tissues. Yeah, especially the tumor samples. Thank you so much for the question.
Yeah, David, go ahead.
I had a question for Patricia. So you were mentioning you added this histology service to your SRL, but that's, it's a very niche-y kind of training that's required, and I assume you didn't have background in that. So how did you get your person you brought in to do that up to speed if they didn't have that background already? I'm just curious because that's pretty foreign to us in flow course.
I made friends. So there aren't any other research histology cores in Boston except for at the Koch Institute at MIT. And luckily they're across the street from us. And so I made friends with Kathy, who coincidentally lives in the same town over from me. Like, we're buddies, which is weird because we're, an hour and a half outside of Boston. So it was really random. But we're all learning together. So whenever we encounter any problems, we email Kathy and be like, hey, how would you process this tissue, right? So this week we're processing mouse brains. We haven't done that before. We emailed and said, how long do you leave it in fixative? All this other stuff, right? And so we have our little network that we have in Boston, but also our FAS from Leica has been really amazing, and he likes us a lot. And he's been using our equipment to kind of test things on his own, because we're, small and we're not very busy yet. And so he can come in and test his protocols that he's doing for other customers for Leica. And then we just bug him to be like, oh, hey, you're cutting eyes today. Like, can we see you cutting mouse eyes? Like, we want to learn how to do this too. So it's really just been learning on the fly and leveraging the experience that our vendors have, and, just being really nice to them so they want to come back and train us on how to do things. And then going to these conferences has been really helpful too. My one tech, so I bought a ton of textbooks on histology, which, you need to research and study if you want to take the ASCP exam. And so we've been doing that as well. And so it's kind of how I started in flow cytometry. I didn't really have anyone teaching me to do flow. I just kind of picked up a bunch of books, like Howard's book, which was really great. I started going to meetings and the local meeting in Boston to learn flow cytometry, started hanging out in other flow labs. I kind of taught myself that way to do flow cytometry, and that's kind of how I'm applying histology as well. So make friends is what I would suggest in your area.
Very similar question what David asked, and it actually applies for all of you, right? Implementing a new service, new technology, or a new application you're not familiar with, and how you build that expertise within the team. And I guess it's from pain from the past, where a Helios is both, and you expect that instrument to start generating results, and there is no one that knows how to do just the acquisition or the analysis. So how that's incorporated in the decision of implementing a new technology and making sure you have the right person to do this. That was the question. So I guess you already answered, but asking, as
it's when you, I get your point, when you're implementing new technologies, new workflows on new platforms. And each one of those is very separately challenging. And exactly what Patricia's is what I always use is I'm in a department of pathology. I have my clinical connections for years, so I would called up the histology department and said, "Hey, Kathy, who's that? I'm going to start doing this. Can my person come and hang with you?" And that I sort of say, in the immunology field, that's how many of us learned immunology, is that and I say where we are now, the technology is complex. We used to learn techniques. And I say the difference is because the technology has started encompassing major science. I remember when I first learned the MTT assay, I just went and hung out with my friend for a while to learn how to do that. So that's not a bad approach. You don't want to reinvent the wheel. And we're not experts in everything. So I think that's a good way to do it.
And I just wanted to add a comment for the question Anna had. And it's something we recommend before starting panel design with samples that we know might be challenging in terms of autofluorescence, is the customer to run just M-stain cells. And so you will be able to design the panel based on what you're actually seeing in their samples. So that's the part I would incorporate, just based on sample type, suggesting running at least some samples before starting fluor assignment. And question, sorry. You go ahead.
No, go ahead. Go ahead.
Okay, last question for Anna. You were showing the brightness ranking and the spread characterization that I know for us, we provide that in advance. So wondering for the other instruments, do you do the characterization?
Yeah, another thing that I forgot to mention. Yeah, so the idea, I think with starting have all this material because we have so many different instruments, I think would be very valuable to add in every Excel file that we have that we use to assign these floors, to add a tab for the spreading metrics and another one for the stain index, at least for the main floors, right? And so, yeah, the idea is to, since we are doing the optimal voltages characterization, to already run this spreading metrics added to that, and also the stain index. So then you have something specific for each one of the instruments.
Hello. Are you hear me? My question is for Jonni. In the morning when you talk about the mitochondrial health, are you just did the flow cytometry with that, or did you check the mitochondrial health with the help of the Agile and Seahorse technology? Have you ever tried to combine that idea?
So that is a collaborative project, and we are specifically tasked with the goal of creating high dimensional, high data cytometry approaches. There's also an imaging approach. There's also even a zebrafish approach, because I mentioned these are hardcore genetics people. And then we all meet together and we put the data together and collaborate. The one interesting thing that we've been working on this for now 2 years. We have gotten to the point of being able to actually collect data faster than any other technology. And that's because, one, we understand how to use the technology, and all we've done in the past has contributed to it. And there have been others in working with, because what's happening is a lot of what we're looking at some primary samples, but there's a lot of cell lines that have been made from these patients with known mitochondrial defects. So we're looking at cell lines and we're looking at cell lines in other ways. And they've been looking at functional assays, but now that's why we want to incorporate functional surrogates, if you will, into our panel.
Thank you.
Small comment for Patricia. I had to chortle when you spoke about the people skills of the person you hired. I had a wonderful technician who was lending to something else that you brought up, bought out from under me by the institute across the street because my institute wouldn't do a bloody thing about it. But during her interview, prior to her leaving, I asked about her, kind of filled her out about her people skills. Turned out she was a barista at Starbucks. And I said, not a problem as far as the people skills. She will be able to deal with a customer from hell just fine. But my question also comes, now we've had this conversation, and Jonni and I had it last night. We've had it with Rui a little bit. So I just want to make it all public. What do we do about the support from our institutes when they say they're willing to give an investigator, a new investigator coming on board, a machine of their own in relation to what we think is NIH with rigor and reproducibility, where they're supposed to be going toward cores? How can we try to stem this? Because this isn't going to end well for us if investigators keep getting their own machines.
That's a good question.
So I've done a little bit at Broad because every time somebody reaches out to our procurement department to ask about a flow cytometer, they immediately put them in touch with me. And so something that I have done, and I've been able to prevent people from buying their own instruments, is going directly to the PI and be like, what's your pain point here? Like, why are you buying your own instrument? How can I help you? And it might be as simple as, well, we need to do BL2 plus work and you don't do BL2 plus. And so I'm like, great, I can put one in your lab. I'll take care of it. Or, so I've been able to come up with creative ways, but basically it was me making friends with the head of procurement. And anytime anyone reaches out, I get looped in immediately, right? And so I think this is what we have to do at our institutes. Is that, hey, if you're going to support me and you want me to support myself, you have to stop letting other labs buy their instruments, because you're basically taking away from me when you do that, right? And so if you're going to do that, great, then subsidize me more. So these are the conversations we have to have. Yeah. And yeah, so they don't want to subsidize us, right? Like, well, they are now for me because they have to. But those are some things that I have done. And I think that has really worked out. And also working with my vendors. Like, I don't want my vendors going directly to my researchers and saying, oh, you need a quote for this? Let me get it for you. They come to me mainly. Most of them do, not all of them, but the ones that value our relationships, they come to me first, or they loop me on the email and say, hey, by the way, you have a core facility, did you know that? And so I've been trying to make those relationships. So that's one thing that I've tried to do.
So it's a little bit different, but the same basic idea. So at a university, in our university, there are many schools, but there's only one core that covers all the schools. And that includes engineering, that includes the vet school, that includes all of those. So that was able to be done over time. And I basically say we kind of earned that over time. And these labs, we had demonstrated expertise. And I remember going to talking to a big name at our institution in early days of gene therapy, Jim Wilson. And he was going to buy his own cell sorter. And I basically said, I think you probably don't want to do your own cell sorting. You want to have cell sorting done, but you don't want to do it. Well, we know really well how to do it. And we have developed a model that brings us in between $500,000 and $700,000 a year of kind of doing what Patricia said. Big users who don't want to share we have high support from our dean of the medical school. So who basically get an instrument and then pay us to take care of it, train them, set up our information systems and all of that. So yes, and we have so many different types of instruments. They don't really use all of their own, but they pay us for, again, what I think we need to sell more, and I like the way you're doing it, is our expertise. Because instruments, by and large, are getting easier to run. I always say, I always have high school students in my thing. They can run instruments. They can't necessarily set up the exchange, but they can run the instruments, and that includes cell sorters. So we are the experts in the field, and the more we develop that, the less it is enticing to other groups to have to hire somebody and pay, which they're not going to be able to find nowadays, because I love that last slide where everything is real. And really everything comes down. A lot of our problems now is staffing. So remember, we know what we're talking about, and we continue to learn. So we will continue to know what we're talking about.
And I think that's really important, as Jonni says, that you go to the PI and say, look, if you don't have an instrument that's really well taken care of, because it's not being taken care of by your lab members, your data is going to be really terrible. And this is one way I was able to convince a small group at our institute that they had an Accuray they had not taken care of in like 5 or 6 years. I think they were just analyzing bacteria, honestly. And it was in a CRISPR lab. So they were the genetic perturbation platform. They were making CRISPR cell lines. And I said, can I please put a Cytoflex in here for you? You'll get trained in my lab. Everyone will get, because they're a BL2 plus lab, right? They were working with lentivirus. And I was like, everyone gets trained in the main lab. You'll get access to all the ones in the main lab in case you need them. Plus you get access to the one in your lab. And we take care of it. So that instrument isn't as high used as the other ones. And so we probably QC it like 3 times a week versus every day. But they know that it's working when they go up to use it. And if it has a problem, they know who to contact. And any new lab member gets trained by us and not by whoever happens to be in lab that day that's available to train them, right? And so it's reliability, it's reproducibility. And so these are the things that I sell. And I've only one time not been able to convince somebody in our cancer program, so they bought their own Cytoflex, only because I got them such a great deal that I almost shot myself in the foot, right? But guess what? It never works. And it's only a 1 laser Cytoflex. And I'm just like, so they're still using ours. But every other time I've been able to bypass work with put an instrument in. And I've done this for cell analyzers and sorters. So I have 3 analyzers now in satellite labs and 2 sorters. And now I'm thinking about this for our new building that's going up a few blocks away too. So, and there's other equipment. So I want to be the person that people want to reach out to if they need new equipment too. So that Luna 4, the Comet instrument, I basically said, someone came to me and said, I don't have any funding to buy this. It would be really great if we had this at the Institute. And I said, guess what? I can't buy this instrument this year because it's not out yet. I still have money. I'll buy that for you. And we have mixed models of operating it. So they do it as a full service and I do it as training. So these are just ways, like any way I can help you, I will try to help you. And I think people value that too. If they see that you're not trying to come in and tell them what to do, and you're just like, hey, how can I help you? And I think that goes a long way.
Yeah. Oh yeah, go ahead.
One last comment. I basically think all of us, one of our, if you will, mottos and driving forces is providing access to the technology to investigators at any level. An investigator should be able to come to us and say, I want to do X, and not know anything about it, but be able to leave and be able to do that. Or somebody can come and say, I want to do X and I think and so I'm going to buy this instrument. Well, first of all, one thing that changed at our institution, and that is no recruitment packages from the school can purchase core instruments. They can use it if they want it, they can use part of their recruitment package, but it must go in the core. And it must be available to everybody or in some ways. So a lot of, we have 9 satellites. I guess the biggest one is Carl June. Carl June could afford to buy any cytometer he wants in 20 of them. But many of you know, he came from a cytometry background. And if you've ever done calcium flux, before he did cell therapy, he was doing that. So he appreciates what we do. And somebody asked him, well, why do you still work with the core? And he said, why wouldn't I? That's one less area of expertise. I have to find people to do it. So that's what, multiple levels. It does help. We have central support for this, but it's mainly getting people to appreciate our expertise and what we know, and that their work will be better because of doing this.
I just want to add, I mean, those are really great suggestions. But there are also other factors that are really important, like the culture of the institution, right? So for instance, Tricia was saying that she has a good relationship with procurement, and so they enable her to understand who's trying to buy instruments and things like that. Or for instance, when you ask a lab that, you have expertise, or we have expertise, and I've asked certain labs, so why don't you use the core? Why do you buy an instrument for yourself? And they say, well, we don't need your expertise. We are experts, and we can ask the institution to buy the instrument. So we're going to continue buying the instrument. They don't pay service contracts and things like that. So we have had to have a lot of creative solutions, like we service other labs, and you have that experience, right, Jonni? Servicing other labs that have their own instruments. So we started a service to basically, create a bridge between those labs and the core, because once they have the instrument, they never go to the core unless the instrument breaks down. And then they come.
Entirely.
Right?
Actually, so part of it is, yes, what I'm talking about, our model's been in operation for many years, but we didn't start that way.
Right.
It built to that and expanded. And for instance, there was somebody in the Department of Cancer Biology, and they really wanted a fusion. And this guy wanted a fusion primarily because he does clinical trials and he wants to be able to do things at the drop of a hat.
Mm-hmm.
So we can pretty much do that. But his chair said, no, you're going to do it with the core. And he wasn't happy, I will say, but he did, and he uses it about 10% of the time. That's all he uses it. We get to use it and bill for it the rest of the time. So we have different models like that. And I think I think that's critical for us in big institutions, because it's also better. And our school is making this, as I said, new policy that instruments, they don't want redundant instruments, but for reasons you might not guess. And they can even be small instruments. And it's because of space.
Mm-hmm.
I do not need 10 things up there. If somebody's using this dissociator here, and maybe it only costs $25,000, then we're not going to put one next door, because, we're an inner city campus. There's only so many places to go. So they see this at the institutional level now.
And I have to say, I put dissociators on carts and I wheel them around the institute.
Right.
I have 3 of them. They just wheel around. Yeah, you just check them out like a library book. So I have 3 Gentle Maxes. I have some little sorters on carts, like the Namocell. Everything if it can wheel, it's on wheels.
Yeah, we actually thought of that when we were testing the Wolf and thinking of buying it. We were thinking of. Yeah.
I want to say our dissociator is mobile, so it's fine. So I am actually from BCM Core, so I just have a question regarding your backbone panel. So we do have sort of MIPS, the mouse immunophenotyping panel. So I'm just wondering about how much is like how put together is the service, right? So like, hey, this is the panel. You go ahead and buy it from BD or whichever company, or like, hey, we have these things, antibodies, like as a bank. Like, hey, I can tell you the dilutions. Like, how far is this as a service being offered? My question.
Yes. So for the backbone panel, we actually had to select, right, the ones we want to try, and that's why we're talking about having this good relationship with the vendors, right? Because like I mentioned, this is the second version of the panel, and if you start doing this project and maybe you don't have the budget for that, this can really help, because most of the companies, they have the sample site, right? And they can send it to you and you can try different floors. So this was kind of what happened to us. Like we designed, I don't know if you noticed, but most of the backbone is in the VUVs.
Mm-hmm.
Again, we started this 1 year ago, and we didn't, I think even there, I don't know if Thermo was doing BUVs at that time. Now I think they are doing. So it was a struggle to find like alternatives for that was not BUVs. I think now we have more, and it's kind of trying to intercalate in them and say, okay, we have this fluor in this and we have this fluor available in that. So for the backbone, because it's something that we are putting together, was our choice. But when we are designing a panel for the users, sometimes they already have in their lab and they are happy with that. So we just try, okay, so now you just move this to this and that, and these are the alternatives that you can work with. And most of the time we try not to give them like the catalog number. We just say, okay, this would work in this position, but they can buy for any component that they prefer.
Oh, so it's not like how we get like a kit saying, hey, this is available here. I just
no, yeah, the idea for the backbone was not like,
it's just like a guide saying, hey,
the backbone per se, like we want to publish, and then of course, like any OMIP, you're gonna have the catalog number, the clone that we use as a guide, right?
Mm-hmm.
But yeah, that doesn't mean that you can adapt.
Gotcha. Thank you.
You're welcome.
Yeah, it's very important to add that a lot of the work, and that's why Anna's saying that we started it a year ago, because a lot of the work is actually done by our researchers. So we're not staining the panels most of the times, and they're actually testing. Somebody comes with a similar panel. We give them the backbone panel and a few more add-ins. And so basically that's how we're testing and getting results from our research.
I think, like I mentioned during the talk, it was a lot of different users come often and ask for the same markers. So every time I was redesigning the panel, right? And then of course with that, we're also learning and say, okay, this worked really good in this color. Oh, this fluor is really good for this mark. So it was kind of this, was not something when they wake up and say, okay, today we're going to design this. Was gaining experience with the service that we are providing. And then say, okay, now we know which markers are more important for most of them. Now we know which fluors are working better for this and that. So it was like a experience that we're gaining with time, right? It was not just like, okay, let's do it.
And we don't charge for this service to build panels, because that's also coming back to what we were talking about. It's also a way to attract a lot of people that have their own instruments and want to build panels, and they come to us, they see that we have the expertise, and they start using our core facility way more.
That's something that makes me nervous. This is a whole lot of time from people in your core, but you don't charge for it. Somebody's paying for that. Your other users are paying for it. How do you justify that?
Right. So we basically, the way we set the prices for the instruments, we are including troubleshooting time, not only of the instrument, but also of their experiment, right? If they decide to use it, great. If they decide not to use it, then it's on them, right?
So they are paying for it.
Essentially, yeah.
Okay, so you're just saying you don't do a separate
Exactly.
charge. We do a separate consultation charge for
Right.
that. And our instrument costs, it helps us keep some of those down so that the people who need it are paying for it.
Right.
And not. But either way, it comes
Yeah.
out the same in the end.
So we thought a lot about that, the pros and cons. The thing is, if we start charging for that service, then people don't come to us, especially at Sloan Kettering with the culture that they have. If they have to pay for something, they'd rather not do it. So we want to make this a very collaborative endeavor, right? To really to get people to come to us so that we can help them. So in a way, I think for the culture of this institution, it works better like that.
And I would say that it's a win-win situation as well, because sometimes the users, they think they know until they sit with you and say, oh, wait, why are you gate like this, right?
Right.
Oh, I didn't know that. So I think it's a way that we show like our expertise. And then they start, okay, yeah, I need the core. So they start like coming back much more often than they would do if they thought that, okay, I'm good. I don't need them.
I actually remember discussing this with Joanne Lanigan because she also charged, for consultation, and she said that people would try to grab her in the corridor or when she was going to the bathroom because it wasn't counting, right? And so she had to say, no, we have to go to the office and talk there.
We don't go to that extreme. We have sidebars and those types of things.
Right.
But if someone comes, you're talking a whole panel development
Right.
process, we estimate, because if we're doing that, then we're not running samples, we're not sorting cells. So it becomes a loss of money.
Mm-hmm.
The idea is that it's going to change, it's going to generate money down the line.
Right.
But this way, and if we add it all, because we do a cost analysis and we charge what we have to charge to cover our services, then other people are paying. But we frequently, people will call up and ask a question. We're not lawyers. We're not keeping lawyer logs. We ask. It's really more on a project basis.
Right.
And people who, the first thing I always say is that the amount of money you're going to pay us on the instrument, especially if we're looking at a spectral high parameter, will pale in the amount of money. And I tell them up front, oftentimes, because it's not unusual for us to have people who do major projects to spend $40,000 on anybody.
Right.
To do it, because it costs. A lot of these are special conjugates and things are not routine aware. That's getting better though. I think the vendors are getting a lot more, and there's newer dyes, so I think that's
Yeah, and we rely a lot on, like Anna was saying, on the vendors to provide us samples. To answer also the previous question, where we of course we start getting our favorite fluors and combinations, right, of conjugates of antibodies and fluors. But as new dyes and new antibodies are coming out, new alternatives, we'd like to test them also and see if they're good. So having vendors help us with testing these options also makes it much exactly. Right. Yeah, go ahead.
Great. Thanks discussion. I just wanted to go back to the previous topic because something that is with me, and I really appreciate all the feedback that you guys have provided, but I just wanted to see whether mainly like you, Patricia, are willing to share like the strategies that you use to be getting 3-4 instruments every year, and even like volunteering to place instruments in the single, PIs. I'm struggling just to upgrade my instruments. So is this all institutional support or?
Yeah, so I'm at private nonprofit, which has access to more funds than maybe a typical academic setting. And so I'm fortunate in that I get to request what I want every year for my capital equipment. Whether or not they fund everything is different, but the past few years they have funded everything that I have requested. And so I am highly unusual in that area. I don't have to write, grants to get instrumentation like S10s. I don't have to wait 4 years to buy something, right? But that's part of Bird's mission, is that we are supposed to be on the cutting edge of technology and science. And because of that, they know that they have to invest in instrumentations, and they would rather, because what Jonni said, space is our biggest limiting factor right now. We only have so much space. We can't create new space. Space is very expensive in Cambridge. And so we don't want to have a lot of duplicates of instrumentation, right? So it's better to put one instrument in the core than to have every PI buying their own like high-end microscope or something like that, right? And so I think you can have, I was at Harvard previously for a while, and Harvard had a different method. Again, you could request capital equipment funds through the institute, but the dean had to approve it. And there you got much less every year. Like it was like, maybe you got one thing a year if you're lucky. But you have to have a conversation with, the higher ups at your institute saying, like, this is going to save you in the long run, because you can come up with like, what I'm doing right now is like, what is the square footage of every piece of equipment that I have? How often is it used? Some of the justifications for me getting the histology equipment right, I went around because we had cryostats at Broad. And I said, well, how much is that one getting used? I would walk by and do a census every day. I would see nobody on it. And so it didn't make sense for that one to be there in that lab. And this was just a random lab, but another lab down the hall might use theirs all the time. And so it's like, okay, well, we definitely need them at Broad, but should we have one in the core facility instead? And not have it in an individual lab. So those are things that you can do. You can say, like, look at this equipment is not getting used here at all, but in this other lab it's getting used all the time. And so you just do your own homework, I guess. It's a little bit more work on your end though, that you have to do. Like you have to figure out like how much square footage things are taking up, how high use these things are, how much they cost to maintain. And then you just make all this, like you give this information to your institute and say like, this is the real numbers for how much it takes to maintain this piece of equipment and how many people hours it takes to maintain those and how much, space it takes, because space is worth something at your institute too, right? There's a dollar value on that square footage. So I don't know. That's kind of a scrambled answer, but it's a lot. It's a lot of work. And I've done all of those things in order to figure out like how I can get more funding towards my core versus the PIs getting that money for their own labs.
Yeah, I'll ask some online questions because there are so many actually, but some of which have actually been asked here. But actually, I'll start with a question to Anna, but actually could be extended to also Tricia and Jonni. So one of the questions was, in light of Omar's presentation on Easy Panel yesterday, how do you think the advancement of AI panel design tool will impact this service? And before you answer, I want to just extend the question to also Tricia and Jonni, in the sense, how do you deal, especially you, Tricia, with so many different technologies? How do you deal with disruptive technologies if they come about? Because you have to have a lot of expertise to understand when they're going to be disruptive or not. How do you handle those things?
So it's not so much me handling them. It's more what I see the researchers doing. Right. And so I follow their lead most of the time, and I kind of keep an eye out on what people are asking for. And then I ask, like, why are you asking that? And then I go and do a little bit of investigation, because like, it didn't happen overnight that we stopped sorting cells into microtiter plates, right, for single cell. It slowly increased over time that we just stopped doing that. Right. And I was like, where is this going? And I kept hearing about 10x Genomics, like, oh, so-and-so's lab got a 10x box and she was doing all this work. And I'm like, what is that? I want to learn. And so that's kind of how I stay on the edge of like, what is the new hot thing that people are doing? Right. And also I look at the recent publications that are like bioRxiv or something from the labs coming out.
Right.
We just like there was a talk at AGBT from one of the labs doing slide tag seek, where they're basically tagging nuclei on slides and then taking it off and then putting it into the 10x box off the slide. And so they reach out to me and are like, hey, can you sort this stuff for us? Because we still find a lot of debris in these samples. And I'm like, sort a section off a slide. Like how?
Right.
and so I'm like, okay, let me figure that out for you. So now I'm like trying to figure out like, oh, is this something that maybe a deep cell type imaging sorter can do, because it's microfluidic and it's image based. And so these are the things that I'm trying to figure out how to help people with. And so that's how I kind of stay ahead of it.
How do you find these technologies? I mean, do you Google? Do you talk to people?
That's a good question.
Just comes up?
I mean, I do.
It's me asking.
What? The meetings are good, but I think also because I have so many relationships with vendors, and because vendors hop companies so frequently, they often will go to a new company and then reach out to me and be like, hey, I'm at this company, Tricia, and we think that your technology would be great at your facility. And then I investigate it. I'm like, oh yeah, that's interesting. Or I'm like, so yeah, I don't know. But I think being at the place that you're at also helps, right?
Exactly. Right.
Yeah,
and
but
Chat GBT is like going to take us all over.
Exactly. Yeah. Speaking of which, AI.
yeah, speaking of it, is it panel? I don't know. Even Omar yesterday, I don't know if I was looking at him like kind of angry, and he said, no, but we always need the human part. But no, I think like this is great, right, Omar? I think everything that can facilitate our life, we should be happy.
Right.
So I see this more like a tool that we can use to provide a better service or a faster service for users than actually something that we don't need you anymore. We can do it. But no, I think it's great.
So I'll just finish up with one last question for all of you. How do you get institutional support for implementing major changes in your SRL? Jonni, in your case for clinical collaborations and initiatives, and Tricia, new technologies, capital.
I guess the way I do is somewhat the way Patricia's trying to do it. And that is I go and ask people. I tell them, I talk to them. So one thing that was mentioned, how do you get instruments? I have currently 42 instruments. I have one of them was purchased by an S10. None of them was purchased by the institution, meaning central institution. So my usually that I've developed for years has been going to the chair in X department that someone really wants, and I say, okay, here's the deal. You pay this part, and with the number of instruments I have, and I'm allowed to keep my depreciation in my lab, I get a pretty substantial, probably about $300,000 to $400,000 a year in depreciation. So that can buy an instrument almost. But I'll say, I'll match this, and your new recruit will have access to the instrument. Now, the first thing people say, okay, then we get to use it for free. No, NIH rules do not allow that. You cannot everybody has to be charged the same amount. What I do tell them is that, well, if you want your investigators, I just did this last week to the engineering school. In this case, it was an S10 instrument, and the engineering school was new to this, and their investigators thought the instrument was theirs. It was located there, but it wasn't theirs. And the chair said, how do I explain this? And what can I do? I said, you can subsidize your faculty's use. I charge everybody the same. You can pay for it. So I offer them different alternatives. But the fact is, you really have to use everything. I've gotten some, and Broad probably can even do this if you haven't done it, because it's a nonprofit, as we are. But donations. So most grateful patients, which are the biggest source of donations, go to will go to clinicians and say, I want to fund something for you. Well, I've talked a lot to the clinicians who we do a lot of work for and all, and very frequently they will recommend, and we're perfectly set because people like things. They like to have the Susie W. Smith Memorial Flow Cytometer. And it's funny, but it's true. They'd rather have that than the conference room or something like that. For some reason, they like things, but we don't have access. So that's a good thing. And the other is basically, we've gotten about 4 instruments from doing partnerships or work with biotech or pharma. GlaxoSmithKline bought me a sorter if I would agree to train their people, for which they pay. Okay, but they gave it. They could get it. It was a donation for them, so it was beneficial. So nothing's off the table. You have to get creative funding.
Yeah, that I do a lot of that as well. So I go to the PIs. I ask them, and a lot of technology actually comes out of the Broad as well. So we have a lot of startups that spin out of Broad. And so some of that is coming back to Broad. And so it's really a lot of just engaging with the community and knowing like what they need. And they're your biggest advocate sometimes for getting things into your lab. So if, so-and-so PI needs this because they just got it in, a giant multi-site grant from the NIH to do a project, then you're going to get that for them. And if you partner with them and say, hey, like, let me help you with that, then they're going to put it in the core and then everyone has access to it as well. And I think those are, the best relationships that you can foster. It's just, and a lot of my, the junior PIs at Broad, I actually sorted for them when they were grad students at Harvard. So they owe me. It's coming full circle.
Okay, I guess that's a wrap. I want to start by thanking the speakers for their wonderful lectures and this roundtable. Thank you very much. Thank you guys and our online attendance. Also, I just have 3 announcements, right? So tomorrow we start earlier at 8:30, right? The other thing is if you want to validate your parking ticket, just come to us. And don't forget that tomorrow you're supposed to use the UT Health parking lot, right? For validation. That's it. Thank you.